please. Okay, the work package 9, it's a joint research activity. Yeah, the screen is being shared. It's on the development and validation of advanced approaches to the monitoring and evaluation of clinical and pathological outcomes of experimental infections. It's a long title. It was a challenging work package. Next, please. What we worked on, was to develop and implement ways to study clinical and pathological outcomes of experimental infections at the physiological, mechanistic, and molecular basis. And with conquering the challenge to do that under high biocontainment conditions and the species of interest, which is in scope of VetBioNet, also including farmed animals and wild animals. The task 9 was to develop the processing of devices and specimens. We'll have 2 talks on that later. The validation of physiological and behavioral monitoring, in vivo bioimaging and in vitro bioimaging. Next, please. On the task 1, the processing of devices and specimens. Main topics, the decontamination strategies of samples to be analyzed, which was impossible to validate. And secondly, to sterilize or decontaminate materials and devices that had been in contact with pathogens. And we have 2 talks on that topic. Firstly, on in vivo imaging presented by Josiane Teuns from our institute. We are implementing that in our new BSL-3. And secondly, the repeated decontamination of a confocal microscope, work done here in Tours by NRI together with Leica company. So can I please have the first presentation? All speakers were asked to allow for some questions within the time slot of their presentation. If that fails, we move on to the next presentation. Josiane, please.
Thank you, Maartje. So this is for later on. Presentation. I think it was the bottom one in the list of presentations.
You have to tell me the name and otherwise
it's the bottom one.
This one?
Yeah.
While you're switching, can I just? I'm sorry, I'm just quickly interrupting. I noticed that Albert is connected online, and Albert, can you open your camera? We just want to say hi, and that we actually miss you quite a lot. It's a pity that you can't be here. So I understand that you will soon go on retirement. Albert, can you hear us? Perhaps he's in his pajama. I don't know. No, he doesn't want to respond already in return. Perhaps later. Okay. Again, Albert, you're missed very much. We're having a good time here. It would have been a pleasure to have you here also, and we hope that our paths will cross again. Sorry.
Okay, so I'm going to give you a very short update on what we're doing for developing a containment box for imaging. Can you show the next slide? Yes. So at Erasmus MC, we have brandly new BSL-3 facilities, and we work mainly with smaller animals. So all our animals are housed in the isolators, And smaller animals are housed in cages in the isolators, and ferrets are housed group-wise in the complete isolator. And all animals can be handled when they are anesthetized. So can you show the next slide, please? Yes, and we do have imaging equipment available in the facility. We do have an IVIS spectrum and a micro CT, and these are in the imaging lab. So we need to transport the animals from the isolator to the imaging lab, and for that we need a containment box. So within Erasmus University, we work together with different departments to develop a containment box. And for the imaging equipment, we got technical support from PerkinElmer. So next slide, please. The containment box had to fulfill some requirements because it's the primary containment when the animals are transported and in the imaging equipment. So it should be airtight and leak-proof. It shouldn't be shattered when it's fallen. And the material also could not interfere with the quality of the imaging, of course. And in the end, a decontamination method must be there that also didn't interfere or react with the material the box was made of. So in the end, we developed an 80mm bore cover for mice at this moment. And that contains just over 0.5 liter of air in it. And that's sufficient for up to 3 mice to be in the box. And we can use that box because of its shape, the round shape, for both IVIS and micro CT. So next slide, please. Of course, the animal needs to be anesthetized in the imaging equipment. And for that, we tested the protocol. And the mice were sedated in an induction chamber. And then when they were asleep, transferred to the containment box. The containment box was filled with isoflurane also. And then it was closed and the animals were transported to the imaging lab and imaged. And they were sedated for over an hour. So that's in the box. So that's sufficient. There's sufficient anestheticum in the box for the animals to be under for about an hour. And that's long enough to use the IVIS and also to use the micro CT. So there's a stable sedation for, well, up to 1.5 hours. And that was the end we tested. They probably stay under longer, but this is sufficiently and at conditions that support normal physiology. And for the future, because the animals were deeply asleep now, and that reacts with the breathing frequency, we are, well, trying to have the anesthesia a bit lighter. So next slide. That was it. Are there any questions? Yeah.
Hi, this is Julia from Irta Cresa. Do you have also, because I guess here you cannot feed hamsters, for example, do you have also similar device for?
We're actually working on that. And the first question was for mice, because that's the main research questions in our facility. But for hamsters, it's next. And that's especially to look at the lungs in the COVID studies. Yeah. So that would be the next one. And there the bore cover is slightly wider because the hamsters, well, don't fit that well in the mice containment box. Yeah. So we need to develop a bigger one. Yeah.
And then I guess the transportation is short because yes. I guess in this chamber it's only isoflurane but without oxygen.
It's a combination, isoflurane and oxygen.
Okay.
And that works very well.
Okay.
Yeah.
Okay, thank you. I think now Albert is here if you want to say hello, at least at the end of the question.
Albert, yeah, great, great. We see you now.
Hello, everybody.
Hear me earlier? So it's good to see you. Well, we're sorry that you can't be here. You have been such a committed partner in the project, so I thought we should just say hello and thank you for all your work and wish you all the best for your retirement.
I thank you. I thank you very much, and I think I will miss all of you since I've been working with all of you since the Nadir. So
yes, right.
Has been a very long time, and Irta Cresa was very much committed to the Nadir and to the VetBioNet.
Absolutely, so, absolutely. To all respects, be it through research and services, you have been a very busy service provider also in VetBioNet. So thanks again for this. I think you were on the second rank, if I remember correctly, when it came to services. So that's already quite good.
Well, it doesn't matter what rank you are. The important is to do what you can do and to do the best as you can.
Absolutely. And this has been done. Thank you very much, Albert.
Okay. Very good continuation. Bye. See you later.
See you later. And I actually have a question also. So we were, Julia was talking about hamsters. I was wondering, can you go up to the size of ferrets also?
Yes, that's the biggest bore cover that we can contain in the micro-CT, but it needs to be very long in order to have sufficient air and anestheticum inside. Yeah. Yes, but they can be pushed aside. Yeah, Norbert.
Norbert Stockhofer. The material you're using, does it interfere with your measurements, or is it or are the used beams not that do not interfere with the plastics?
We used perspex, and because of the cylindric form, it did interfere, but we can correct. And at first there was a table in it on which we placed the mice, and that was from black material, and that interfered too much. So we reduced the size, the diameter of the cover. So the animal's now, well, on the floor, and it doesn't interfere that much. It works pretty well. Last question?
No questions anymore, running over time.
Yes, Matje, you're too fast. It's Jan Langemans, because just a quick remark more. Or a question, did you ever have contact with what's it nowadays? PHC England, UK Health of UK, what is it? Health Security Agency, whatever, in the UK, in Porton Down, because they are transporting non-human primates in this way, so much bigger, from a high security environment to imaging facility. So they have all the equipment and expertise to do this.
No, we haven't, but we certainly will now. Thanks for the suggestion. Okay, thank you very much.
Right. Next talk is right from here, from Tours. Mikael Rieu is here. He has done the work on the decontamination of the confocal microscope, an old one provided by Leica, to see what impact decontamination of that equipment would be. So Michael, welcome. You're so good to be here to answer any questions. And you asked me to present the slides on your behalf. So please, next. Next slide. Oh, you can move. Okay, good. Okay. Formaldehyde is almost a taboo in Europe now, and replacement alternatives include hydrogen peroxide vapor decontamination. And it can be used for level 2 and 3 laboratories, please. And all kinds of research is conducted on pathogens, for instance, host and pathogen cellular interactions. And that brings into scope, of course, the confocal microscope, please. And with the risk of possible contamination of the equipment. Next, please. So you need to decontaminate the equipment in case of maintenance, in preparation of maintenance. And the objectives of the experiment were to analyze and validate the inactivation of pathogens and the functional impact of hydrogen peroxide vapor on a confocal microscope from Leica, but the brand doesn't matter. None of these confocal microscopes are designed to be decontaminated. They are worthy to be used in high containment biological studies because you can have really high resolution images with fluorescent labels that inform you about intercellular and high resolution processes in cultured cells and also isolated cells. Typically, the lifetime of such a microscope would be 10 years. And if you model for a number of maintenances and maybe some bioincidents, you could think of 2 decontamination cycles per year. So the hydrogen peroxide, which is the method of provided by BioQuell. Is that correct? Yeah. Yeah. Yeah, I think that's the next slide. This is the gassing process. This is the setup in the laboratory, and the robotic thing on the right is the hydrogen peroxide vaporizer. Okay, next, please. There were bioindicators attached in a number of places in the room, also places that may be hard to ventilate. And there were 3 types of bioindicators, being the spore. Yeah. And this indicator is corresponding to the environment of microscope. It's located in ventilations and to the different parts of this laboratories. Okay, next, please. So this is the physical damage to the microscope after hydrogen peroxide fumigation. And in fact, as I said, those microscopes are not designed to be fumigated. Next, please. The way bioindicators were applied was first the commercial Geobacillus steatothermophilus, which is a bacterial spore that can be cultured at 57 centigrade. And in the absence of contact with hydrogen peroxide, in this case, or no heating, it will grow, it will develop from a spore to viable bacteria, seen on the right after 24 hours of culture. Next, please. Then 2 other bacterial specimens were homemade to serve as complementary bioindicators. One is a gram-negative, Salmonella abortus ovis. The other is a gram-positive, which is Staphylococcus hominis. And those were exposed during the same cycles and cultured afterwards at 37 centigrade. Next, please. Precision also indicator to positions of salmonella or staphylococcus, similar for 6 indicator of georescus, same place for two, in particular in the platform of microscope. It's a principal risk to contamination. Okay, so what you see here is that the unexposed indicators all grew, and all the bioindicators didn't demonstrate growth after exposure to the fumigation. Next, please. So there was damage to the confocal microscope and a significant alteration of the performance, which is an understatement for damage to the performance. Next, please. And that tells you that the decontamination is efficient from a decontamination point of view. But next, please. The repetitive decontaminations progressively affect the performance of the equipment. Next, please. And supplementary technical maintenance is needed to compensate operating drifts. Briefly, the things need to be repaired and parts need to be replaced, which is, of course, a pity. Michael, will you be taking questions? Norbert.
It's of course a pity that you cannot use this material or these machines in the BSL-3. Did you think about together with Leica on how to protect the machines? Because in fact it's not so unexpected that certain materials of the microscope are sensitive to the treatment. Did Leica think about possibilities to protect the on forehand sensible parts of the machine, of the microscope?
It's possible. It's conditions. The microscope. It's the end
just
of life.
take a microphone. Otherwise, I don't hear.
Life of microscope. It's to elimination the microscope after experiment. It's ends of life, and to protect the system. It's verificate that this peroxide to penetrate into the microscope and the laser, and to verify this performance it's not altered, because the problem of contamination of microscope it's the platform and objective to ensure too simple to contaminate to introduce in this objective, and it's necessary to the peroxide to penetrate into the microscope and different system for a good decontaminations. It's necessary. It's legislation of decontamination for the French systems. It's necessary to prove the microscope it's no risk for the Leica manager to control the system. But this system, it's extreme conditions.
Hello, yeah, Ryan Waters from the Purbright Institute. I was just wondering, the decontamination strategy of the VHP for the microscope, what circumstances would that be used? Because, for example, is there a way to avoid that, such as using primary containment? You know, either if you're doing live cell imaging, or obviously prior inactivation and, you know, primary containment when looking at slides, to try and avoid the actual initial contamination of the microscope in the first place.
Is the question clear? Let me summarize. If you have your sample in a box that is not spreading anything to the environment,
don't have the sample to so presentations have to illustrate the effect of imaging in not containment sample. It's indicator to me to release, to deposit the concentration of bacteria directly of coupon, cupule, je sais pas, indicator, and to put into the microscope, not its real sample, biological sample. It's mimicry the system for contaminations. It's possible to read the presentations.
Okay, Michael, thank you. Thank you very much for being here. Yeah. Oh, you want that picture? Okay.
It's simple to illustrate for the impact of.
Yeah, but
it's a plate. It's not animal cell.
I'll run you quickly through the rest of the session program to see how things link together. Evidently, the presentations just before were extremely relevant for adapting methods for high biocontainment and also molecular imaging, both at a macroscopic and a microscopic scale. Okay. Next, please. Okay, next, please. So these were the talks we have just heard. Next, please. And Leslie Vedder from Nolde's company will run this topic, but before, next, please. Next slide, please. I want to refer to the TNA presentations we saw, in particular, the bottom one. We had presentations on Monday, really nice outcomes of TNA. And actually, the general impression was that the project served by TNA had a much richer scientific background than we had initially perceived. And the bottom presentation is on Mareks disease virus, which is a herpes virus in chickens, and imaged in vivo. And the comments were that nobody had a clue that in fact they were shedding so much virus from their feet. So it teaches you something. Next slide, please. The next talk will be by Ryan Waters, also on behalf of Tina Dalgaard from Denmark, who managed to be here up and including the social dinner, but had to travel today. So Ryan, please, can you take over and then hand over to Leslie for the physiological and behavioral monitoring?
Yes, it's
Is this?
no.
Okay, thank you. Thanks for inviting me here today. My name's Ryan Waters. I work at the Pirbright Institute. Just going to give a disclaimer. I'm not an immunologist, so I'm the named veterinary surgeon at Pirbright. So I'm presenting this on behalf of other people. Tina has Marcia said, and also research groups at Pirbright who've been working under this grant, under this task specifically to achieve the results I'm going to summarise now. So let's see if this works. Okay, so this within the work package 9, task 9.4, this just to set the scene, I guess. The bit highlighted in yellow here is the activity that we've been working on in collaboration with Tina as well. Obviously, COVID made things a little difficult, but we continued that discussion. So the results here are as a result of the work at both institutes, but also the collaboration that we undertook together as well. And the focus of this is the use of molecular tags to look at the host pathogen interactions, ultimately at level 3, so class 3 viruses. The approach that we've used is to develop monoclonal antibody panels and to explore those with a view to publishing them. I'll come on to that in a bit, to look at the immune responses and to phenotype the immune cells in the livestock species. The aim was chickens, cattle, and pigs. We've managed to achieve this deliverable, I guess, with cattle, and Tina with chickens. We haven't been able to do the work in as much detail with pigs, but there has been some work with tetramers, but we'll speak about that in a moment. So this is just highlighting actually what I've just said. So with the cattle, with a bit more detail here. So both B and T cell antibody panels have been developed, and they've both been published. I'll come on to that in a minute. But one of the second, so the T cell panel, was published in Cytometry A, which is many of you may know it. It's a journal where standardized antibody panels are published. It was the first time actually that a livestock panel has been published in this journal, and the B cell panel before Christmas and the T cell panel a few days ago, which are great things. Good success, it reflects all the work that's been undertaken and the discussions that have been had. And it's a view that the aim of it's an open source journal, obviously, and the aim of the cytometry A is to try and standardize the approaches that are used for flow cytometry, to try and give standardization to panels so that there's more ability to be able to cross-correlate results and communicate results. So once there's a fair bit of work that the postdoc undertook to get those panels in place. Then with those panels, we've then used those to apply to certain experimental settings. The analysis of that isn't complete yet, but the results have been generated. So the first of those is to use those panels for sorting and for subsequent transcriptomic analysis, largely looking at the transcriptome in the B cells. This hasn't been done with the T cells. And the second is application of those two panels to a vaccine study. So it's very preliminary results. It was one study, but that was applied there, and it gave some very interesting data that needs to be followed up on. And with the pigs, there was a development and utilization of a MHC1 tetramer to follow the CD8 T cell responses in an experimental flu model in pigs. And this paper was submitted in December last year. And again, this was supported by the grant. And then finally, the work that I know even less about is with chickens. And that was work that was undertaken by Tina, as had been mentioned, and that was looking at the T and the B cell panels in chickens with a view again to develop these omips to to undertake the same analysis as I've described for the cattle. One of those papers looking at that's been published, and one's ready for submission. And again, for the similar to the cattle, Tina's applied these developed multicolour panels to in vivo studies, of which a paper as well summarising that's in preparation. So this is just describing what these OMIPs are. That's what it stands for. This optimized multicolour immunofluorescence panels, and they, as I say, that it gives an anchor point and a way for people to be consistent in their analysis. They exist, and there's lots of them for humans and mice. As I say, this is the first time that a livestock one's been published, and they're all published in this way. So this is the 85th OMIP that's been published, for example. So this is the one from last year. You'll be able to read the details in the paper, so I don't want to spend too much time on there. And also because I'm not an expert in all of it, but there's been multiple readily available monoclonals that have been sourced and multiplexed together in order to come up with the OMIP. It's one of the prerequisites for this for these publications in this journal is that the antibodies have to be freely available. They can't be, specific to institutes, because of the fact of the view that it's to be used to try and harmonise and standardise the flow cytometry analysis. And so here it just shows a gating strategy for flow cytometry. I don't know how many people are familiar with it, but basically you have these immunofluorescently tagged antibodies that are mixed with cells, immune cells, usually peripheral PBMCs, peripheral monocytes in the blood, and they're then antibodies that stick to those that, let's say a certain marker, CD40 here and CD14 in this example. You can gate on these. So these are CD40 positive, CD14 40 positive, 14 negative. And these are actually the cells that are being defined in this instance as B cells. So they're CD40 positive, CD14 negative. And then you go on and you can then subdivide these cells using other antibody panels. And this one had 8, I think it was. Yeah, this was eight monoclonals. And so you can have lots of different permutations of those. It's described here. So from these cells here, you can then use a gating strategy to look for all different subsequent data sets and subsets. And so that was the OMIP paper. And then it's just an example of like diagrammatic example of then how this was applied to the B cells. So the B cells were gated on, and then a transcriptomic approach, well, actually this was done via qPCR, RT-qPCR, to look at secreted versus transmembrane antibodies. It's not been published yet. It's still being analysed. But with one of the important things to note was that the phenotype of the cells, as defined by the antibodies, could be there was a reflection of the mRNA levels that are associated with secreted antibodies in those cells, which are thought to be secretory B cells as per the phenotype. So this is an attempt to validate what the antibodies are telling you about the markers on the surface of the cells via a kind of functional assay. That showed a very significant signature. This was the other reason I laugh. This is the other paper that was published just recently, and the journal haven't. When it was published, they put B cell instead of T cell. So we've contacted them to try and change that. So this is 89, not 85. This was published a few days ago. It is T cell. You can trust me. It says it here. But for some reason, this B cell, I don't know why that happened. But anyway, so this was, and again, it's the same approach. I'm not going to speak too much about the strategy with that, but it's exactly the same approach as for the B cells. It was just using a T cell panel to optimize those with the right fluorophores for the right markers, etc. And again, that was then applied to a vaccination study where we could, so this is the day 0 is the day of vaccination, day 14 is 14 days later. And there were changes that were seen this is the B cell, sorry, this is for some reason gone out of order. And this is the B cell assay applied to the FMDV vaccination. And we can see that these are secretory, this is a marker that's associated with secretory B cells. And we can see that there's a change in the proportion of these, you know, from the day of vaccination to 14 days later, and this is plotted up here. So there is a change that can be seen when the cells are examined longitudinally after vaccination, but this is one study, 5 animals. This needs to be repeated to validate it. But again, it's the application, the initial application of these panels to a scientific question. Sorry, and this is the T cell approach. As I said, it's very similar to the B cells, and it's using a CD3 gamma delta staining panel to identify, like we did for the CD40, CD14. So these T cells are defined as CD3 positive, gamma delta TCR negative, and that's the T cells. Obviously, there's a gamma delta analysis as well, but with the T cells, they're then subdivided into CD4, CD8, and lots of other activation markers. So again, in a similar approach for the B cells, you can then subdivide those further and examine each of those individually. So it's quite a complex, once you start to drill down and add more colors, the amount of data you generate and the different subsets increases exponentially. So it's quite difficult analysis to undertake. And this then just shows these panels were also, once developed, there's an initial validation of these using confocal microscopy, because you know, not only would we like to use these in flow cytometric analysis, but also in immunofluorescence confocal microscopy. So this is an initial validation that we undertook of all the individual monoclonals to see how well they react in frozen sections. This is of a prescapular lymph node of naive animal, a bovid. And it's just an example to show that with the T cell panel, for example, there were the 3 monoclonals that reacted in flow cytometry. We need to initially validate that they cross-react in tissues individually and multiplex them together to be able to, again, look at the, start to look at the immune cell subtypes that are present in tissues as well, and where they localize spatially. And it's something that's traditionally been quite difficult to do, and generally the maximum number of fluorophores that could be multiplexed together is, you know, you normally see 3 or 4. And we've been attempting to try and multiplex those higher to 5, 6 different colors using a confocal microscope, similar to the one that was being discussed earlier, the ILICA confocal microscope, with hybrid detectors that allows for less bleaching, allows you to multiplex a bit easier using a wide array of monoclonal antibodies, as well as with tunable lasers, which again is a really important factor to enable us to try and multiplex these further. And again, this is just in lymph node. This is very preliminary data where these were applied, but again, we can see, I mean, these dark areas, you can probably trust me, maybe you can see if you squint, but you know, these darker areas here are the B cell follicular areas, and the T cell areas around the outside. So we can use that you can see here that there's the staining in green, which is the CD3, CD4, CD8 respectively. The blue is the nuclei, and the red is an F-actin phalloidin stain, which highlights filamentous actin, just to give the cellular structure. And you can see the outside of the lymph node here, the B cell areas and the T cell areas. So it's worked on the individual basis. All of the antibodies actually have worked from the panels in the confocal tissue sections, which is great. And then there's an attempt now to multiplex those going forwards. Obviously, the project's finished, but it's real value. So we're following up on this with a view to then applying these to infected tissue to look at what the immune cell phenotypes are like within infected tissues around lesions, to try and get an idea of what the cellular immune response is like in situ as well as in the blood. And one side thing that was noticed that was of interest when looking at the T cells in the cattle that we procure and use in the studies at Pirbright is that when we were initially looking at different CD4 antibodies to CD4, we found, in red here is the positive labeling, and we found that we had good labeling with all of these clones in for detecting CD4. However, in one of the animals for one of the clones, we found there were no CD4 antibodies, no CD4 labeling. So either the animals didn't have any CD4 T cells, or there was an issue with the antibody. And when the controls were reran, we found that particular animal, the antibody wasn't cross-reacting. And actually, when we looked further, there is a paper describing a polymorphism in the epitope where this binds in some cattle, some breeds of cattle. But we found this was, for us, it seemed to be the first time it was described in the cattle that we were using. So again, it was a very interesting finding, so that perhaps to suggest not using certain monoclonals if there's evidence of these polymorphisms being present. So this was a really interesting finding. Took a little while to work it out, and we sequenced and found that, yeah, that was the case. So again, it was an interesting thing that finding that came from that analysis. This is very briefly, this is a slide that describes the work that was undertaken in pigs. So there was a study where pandemic swine influenza was, pigs were infected and recovered with that as a control to look at the immune responses and the immunoglobulin kinetics. And that was compared with other control groups, largely with a view to, the hypothesis was to look to see if a cytokine adjuvanted adenovirus expressed H1N1 vaccine, if the cytokine addition potentiated the responses. So I won't go into lots of detail, not least, as I say, because I'm not too familiar with all of the detail. However, from an antibody response, we can see here that just that the adjuvanted, the cytokine adjuvanted responses, cytokine adjuvanted vaccine prep seem to potentiate the immune responses as per the immunoglobulin kinetics, with the exception of IgA, and the highest IgA production seemed to be detected in the serum after infection with the actual virus itself and recovery. However, the element of that study which was supported by this task was actually the development and application of MHC tetramers to look for the antigen-specific CD8 responses. And actually, the main sort of take home from this slide for me was the fact that we could see a potentiation, an increase in the frequency of these antigen-specific CD8 T cells in animals. This is at post-mortem in animals which had received this potentiated vaccine. So the aim was to look at the effects of the cytokines, but the tools that were used to do that were developed as part of this, were supported by this grant in the pigs. And this is just a summary slide of what I've just said, and the wider scientific question. And interestingly, there was a challenge that was undertaken afterwards to see if there's a heterotypic protection when you, after prior infection and or vaccination, especially the effects of the potentiation with a cytokine to drive this heterotypic immune response. And actually, although this has already been undertaken in mice, and there was seen to be a very strong heterotypic protection, this wasn't present in the pigs. None of the pigs were clinically protected when they were challenged with a different virus. So again, it highlighted this tool that was developed, helped to highlight the differences in species responses to the same construct. And then finally, the chicken OMIP slides that Tina had provided that I'll just present now. So again, it's a very similar approach to what we took in the large animals. The cells were in the chickens. The cells were separated from the blood cells, the same as we did with the cattle and with the pigs. However, there also is some work being undertaken to try to add antibodies and to add this immunofluorescence panel to whole blood. So you don't have to go through the process of separating cells out, especially for chickens where the volumes are quite small. It's even more of an advantage to do that. So that's the ultimate aim, is to use a no lyse, no wash step. So you can add the antibodies, analyze straight away. It's obviously a lot quicker as well. But all the work that I'm just going to present now is all done on separated cells, PBMCs. So this is just highlighting some of the challenges that are present in chickens compared to other animals. There's a very different way that they have the gene rearrangements with the B cells in chickens compared to cattle, for example, as well as the lack of reagents that are present and the different gene subsets. And actually, there's a real lack of cross-reactivity with most antibodies that are present. Some of antibodies that we have for cattle and sheep and pigs do cross-react both with each other, and also some antibodies that are present in mice that cross-react across species. However, that cross-reactivity is a lot less with the chickens. So this is a paper that's published. Again, there's lots of detail in here, but the main approach that was initially used was to look at in vitro stimulated cells. I think this is with ConA. This is then gated, looking at the morphology of the cells and then looking at the subsets using already existing antibodies that work. However, one of the big disadvantages with the chickens for these polyclonal stimulate, these omit panels for T cells, is there's no T cell receptor antibodies that are of a good affinity and reproducible labeling. And then there was some subsequent work that was used to as I say, to look at in vitro and ex vivo lymphocyte activations. Oh yeah, it is with ConA. Yeah, so there's this ConA chemical that was used to stimulate the cells, and then antibody panels that were then used to look for markers of activation. So it was a slightly different approach that was used in this context for the chickens compared to the cattle, just because there's a lot less fundamental data available. And there was an iteration of the labeling process. Different multi-color panels were applied. And there was an initial view to do the same approach, undertaking a development of a B cell and a T cell panel separately. However, due to various complications and lack of reagents, it was thought to be more appropriate to actually multiplex these together. So this is a 9-color panel that covers both T and B cells in one labeling set. And this was further developed to end up with this 11-color T cell panel. And again, I don't need to go through the details, but the same labeling strategy is applied for the gating of the chickens. And actually here, this is where this T cell receptor antibody was used in place of the stimulation and the difference in morphology of the cells. And actually, because of the fact, one of the complications or difficulties is, as I said, as you start to add colors, you exponentially increase the amount of subsets that you can produce. So actually, there is an approach we haven't done, but Tina's undertaken this as due to her expertise in this area, where she's used this is like an unsupervised machine learning approach to define different subsets. So instead of a human having to go through each one and plot it and look at the numbers, you can use machine learning in an unsupervised fashion to look for different signatures and changes. And actually, this is using this panel before stimulation and then applying the panel after stimulation. And actually, you can pick up the take home here as you get these different populations present before and after. And you can see here in this CD25, which is a T cell activation marker, you can see changes in this, which obviously is thought to be reflective of the activation status of the T cells. And then finally, this panel that was then developed was then applied to a IBV, like avian coronavirus study, which is looking at potentiation of a subunit vaccine using TLR and C-type lectin receptor agonists. And there's different experimental groups here. I won't go into detail with them. However, the animals were vaccinated, they were boosted and then challenged with a 10x of a pathogenic IBV virus. And we can see this is the amount of, one of the ways that IBV protection is assessed is to look at the amount of live virus present in the trachea. So this just shows here that in the animals that had this group 5 vaccination, there was a lot less, and the cutoff for protection is thought to be here. And so then the idea was to look to see, can we see any differences between those groups in the immune cell, circulating immune cell populations using this panel, this 11 color panel, both before and after vaccination. Before vaccination, the answer was no, but after vaccination, there was a significant difference in this CD57 positive cell frequencies compared to the first 3 groups. And so there was an indication there that this panel can be used to at least show differences in responses after infection, after challenge, I should say, in this study. But this is further being analysed. But again, it's just to show the application of these OMIPs in experimental studies, in addition to the development of them themselves. So that's everything. I can have a breather now. Thank you very much. So any questions? I won't have the answer necessarily, but.
Okay, I was afraid of battery life here. Regarding time, I think in summary, this is a sophisticated technology that will allow you to analyze immune responses in livestock in a more mechanistic way, and also have a better understanding of the biological varieties that you find there. Those platforms are open source, right? Anybody can use them. Would you expect this also to be applied in veterinary pathology or diagnostics or epidemic surveys or whatever, or just in experimental settings?
So it could be, but I think and people do actually try and use flow cytometric analysis. I mean, not with these OMIPs with these multicolor, this many colors, but 4 or 5 colors to try and look for vaccine efficacy responses, or potentially, doing surveys. But you could use it, but I think that standardizing kind of antibody analysis for prevalence of disease circulation is a lot more straightforward and a lot more reproducible at the moment, across the world. But it could be used. Obviously it is used for some other diseases, such as with HIV infections, etc. T cell populations. But this level of granularity is probably at the moment appropriate for fundamental research. But there's no reason why it couldn't be, I guess.
Okay.
I've been standing in front of that all the time.
Thank you. Audremo from INRA. Thank you very much, Ryan, for this very interesting presentation and such useful tool for the scientific community. I'm just curious regarding the panel that you presented, were all the antibodies used already conjugated with fluorochromes, or did you have to link sometimes yourself? Just curious about your feedback on that very technical and tricky point of view.
Thank you. Yeah, so I didn't mention, it's a good question. They weren't already conjugated. So the researcher, Edward, I can't speak about the chickens because I don't know the answer, but for the cattle, the researcher, the postdoc had to conjugate themselves, directly conjugate them. There was an approach at first to try and use secondary antibodies, but with all the different colors, and basically everything's IgG1. It's very difficult to do that. So yeah, he had to do that. It was quite challenging to do as well. In the paper, it says the approach that he used and the kit he used, and I know that he used different types. I think Lightning Link and another one. I'm not too familiar with it, but he had to do a few first. But he didn't have success immediately. It's not just he bought it, did the instructions, and it worked. It that took a bit of time. So, yeah.
Thanks again, also on behalf of Tina.
Yes.
Now Leslie Vedder from Noldus will introduce and manage actually the next topic, which is behavioral and physiological monitoring. And something special happened during the project because when everybody was sequestered by COVID-19 lockdowns, there was this vibe in this community to find out what people would be interested in, to measure and to monitor and to have alerts in infectious diseases studies in a huge variety of animals. And that has resulted in a very nice presentation at the online meeting of VetBioNet in December 2020. And from there, working groups started under the inspired management of Noldus. And we will now have an overview over that activity, that work package, and also a number of use cases. And for that, I would like to hand over to Leslie Vedder.
Yes, thank you, Martje. Yes, so what Martje introduced, I will explain the summary of what we did in work package 9.2, and then we have some speakers that will tell you more details about specific cases. So the objectives and the project activities that we do, I will tell you a bit about. I will tell you some general results of all the groups that we defined at the beginning of this project, and about future developments and strategy from the conclusions that we made from the projects. So the objectives of the 9.2 package was to develop better tools for physiological and behavioral monitoring, especially the body temperature, general activity, eating behavior, drinking behavior, and also to find temperature responders to get real-time data transmission from the animals. And to also find an objective definition of the scoring of the human endpoint of the animals during these studies. And for that, real-time data was needed, and also to have automatic alerts, and reducing transition of humans in and outside of the facility rooms where the animals were kept. So therefore, also, again, the remote access to the monitoring equipment and real-time data. So because the COVID hit during this project, we had to sort of try out how to manage the groups and to get all the results from everyone. And therefore we organized group meetings, and that's what we have, the 6 groups defined. We had several discussion group meetings. So we had 4 sessions of the 6 discussion groups, and we had 1 combined meeting where we combined all the different groups together, because sometimes there was also some overlap between the groups, that maybe they studied in a specific animal that could also be used in another animal species. And these discussion groups also were really nice, and not all the pilot studies could be done because of the COVID, so we couldn't exchange the equipment, but there were still some studies that had quite some interesting results that were not part of the VetBioNet 9.2 package, but that had nice results to still present in these groups. So these results of not really of the pilot studies, but still the results done by individually by different partners will also be in this presentation. So we had different groups. So we had the ungulates groups that were all part of the cattle, sheep, goats, horses, deers, and alpaca. The pigs, the birds, rodents, carnivores, and the fish. And so we defined those groups at the beginning, but not all the animal species that are listed were part of all the studies discussed. And each of the members of the VetBioNet could register to one of the groups if they wanted to have the information specifically.
The telematic measurement of body temperature. So we, at Nodus, we do not develop this technology ourselves. So we're trying to find vendors that wanted to work with us during this project. And 8 vendors wanted to partner with us. We presented them with a table to fill in for them for the specific groups. And we asked them to, that they have a technology that fitted the arena size of the animals, that could fit the number of animals that we wanted to track, the maximum distance to the receiver, because we wanted a real-time monitoring of the temperature. So the receiver had to be in the distance of the animal pens. The study duration was defined beforehand for the vendors per group, and the measurement intervals. And we had 8 vendors that had solutions for all the requirements. And in the end, 4 different companies were trialed during this project. 2 implants from MK and from TSE, and 2 capsule solutions, and that's from Star RD and Bodycap. So in the end, we did 12, we delivered and tested behavioral physiological monitoring tools for 12 animal species. We tested it at 10 institutes in 5 different countries. We did a lot of testing with different, with both behavioral analyzing and body temperature in cattle, sheep, alpaca, pigs, chicken, hamster, rat, foxes, ferrets, cats, sea bass, and rainbow trouts. And all the technologies that we tried were contact-free and collected remote data. And I will present to you the results per use case, per study group, animal group, in a way of the design acquisition, data acquisition, and the analyzing results. And for the design, it is for each animal group to have a better result with these technologies. It is easier to have a standard animal disease model, and also to exchange the different models between facilities. To record the data in a standard way, to have always the same sort of measurements that are being done. So even if you use different tools, for example, for behavior tools, that you always use activity or proximity with other animals, and to optimize the environment, and also to exchange the lessons learned between the different studies. Especially the environment for some of the technologies can be quite challenging because it can block or the technology. For the analyzing part, new technologies, of course, provide new kind of data. So to make sense of the new data that's coming in from these trials, to have a standard way to record animal behavior provides an opportunity to link welfare-related behaviors to disease models. So you can also then relate, for example, if you know a bit more about the welfare, the well-being, if that affects the disease model. So a sick animal from the start will probably develop the disease maybe differently than a completely healthier animal. More behavioral knowledge in disease models provides an opportunity to determine human endpoint based on behaviors. So the results of the ungulates group. There we did a combination of different technologies. So we did video recording, we did spatial tracking with ultra-wideband to gain the information about activity, and we used body temperature capsules in the studies from Bodycap Medical. And for the restriction was more the design of the rooms. So the rooms are quite small where the animals were being kept, and ultra-wideband tracking is then a bit restricted in this area because you have a really small room, and then you need the precision of the location data, and that will still that needs still need to be optimized for the smaller rooms in these groups, and to get access to the wired sensors. Then 2 of the acquisition was the temperature. So measuring the temperature in real time was a challenge. And this was because the capsules that were kept in the animals the animals were quite big. And therefore the signal of the capsule that was inside the animal was a bit blocked by the animal itself for the monitors. So the animals had to be closer to the monitors to get the results. If the animal doesn't come closer to the monitor, then you don't have a real-time interaction. And it was possible to apply capsules orally in alpaca and cattle. This was tried here. So the ultra-wideband tracking was successful in sheep and cattle. However, because of the different room sizes, we still need to optimize then the settings of the sensors. Yeah, so that was a bit tricky to get the right settings for the sensors in these smaller rooms. Because how often do you want to track it? That influences the amount of data points that you have. Do you need all the data points? More data points can have more activity measured. So these settings, yeah, was quite tricky to get available, and we couldn't repeat the studies several times because of the restriction of the project. And also we didn't have a defined baseline for what is an active animal, what's not an active animal. So that's still something to be picked up after here. Then there was, you have then the ultra-wideband tracking information about the activity of the animals, but sometimes it was still nice to have a video recording to make sense of the data. So you see animals or piling together, or being extremely active, but then it is, yeah, you still need to check or to have a good report of the what the activity that happens in the room. So if a person enters, then maybe all the animals were going to a room, but if only with the sensor technology, you don't have this information. Yeah, and the temperature correlated with the rectal measurements, so it was quite nice. And we did the first study with automatic behavior recognition based on the ultra-wideband tags with centimeter data in cattle. And there we could define walking, standing still, lying and feeding and drinking. But that's still the starting, so that was found, but we need to optimize this model. In the pigs, it was quite a hassle to get the pig studies started, but at the end of the project, we got a pig study started. And because the limitation was in most of the studies that we wanted to test in young piglets, and the weight of the sensors is then a limitation because the sensors could only be attached to pigs on the ear. So if they're too young, then the ear tag was too heavy for the piglets. But we managed to do an experiment for 3 weeks on 8-week-old piglets. So we had a successful study with individual activity based on the ultra-wideband tags. And there was minimal damage to the ears in this study, so it's quite good news. And then, yeah, good lesson learned from this study was to prepare the study was planned 3 weeks beforehand, and there was minimal time to prepare everything, and that yeah, caused some damage on the data. Yeah, and from the results of this study was that activity measurements were in line with the clinical observation. But the individual activity data, yeah, it showed also interaction between the piglets and the place preference that there were in the animal pen, but the location data alone couldn't give extra information about different type of behaviors. So then you still need cameras or something else to detect these kinds of behaviors. In birds, we trialed a new technology. That's Aruka code tracking. So instead of a tag with ultra-wideband, we did a code-based tracking. So then with the camera above the pen, and all the birds were carrying these plates with a code. And we also did a successful trial. That was with the body cap capsules. They were ingested into a chicken's gizzard for a 2-week trial. yeah, so there was continuous temperature measurements in chicken done with the body cap and with Aruco code tracking. Yeah, we need to optimize the plates. We trialed in the brown birds and white birds, and the brown birds, the feathers still overlap in the sometimes over the coat, and then you don't see the coat. Or there were it was in just the home cages of the chicken, and they were not always visible for the camera to detect. And we also tried the Aruco markers with ducks. And in ducks, it wasn't as successful as in chicken because of the wing position of ducks. So they have the wings closer on the back. So it overlaps the coat, so you cannot detect the coat. So the Aruco code tracking was a prototype, and it still needs the further development. But it was possible to detect different behaviors in a novel object test. So we did 20 animal pens with 10 chickens were followed. And we did a novel object test for 5 minutes. So those 5 minutes were studied further with the technology, because during the novel object test, the chicken were always visible for the camera. So you had a better test. And temperature measurements was successful with also correlated with the calorical measurements. And also during the infection of the chicken, you did see an increase in temperature after this infection compared to the control group. In fish, we did 2 different studies. And we did one with sea bass and one with rainbow trout. And the sea bass were grouped together in one big tank. And with the rainbow trout, we attached color beads for individual fish tracking. And there were 2 experiments done, one with individual tracking for different color beads and one with infected fish and control fish with 2 different colors. But still there needs to be some optimization of the position of the tanks, shadows and reflections, so that yeah, interrupts the measurements from the fish. For example, you see here the reflection of the walls. Then also for camera detection, they pick up this as an extra additional fish. Then it was still quite challenging in especially the sea bass room, but we'll get more details about it also after my presentation. Yes, and I want to, so for the acquisition, we were able to get the results for proximity measurements, spatial measurements about the swimming patterns and how close where they were in the tank, they were close to the wall or more in the middle, and social interaction of the shoaling behavior, but also for the specific rainbow trout interaction between the individual fish. And these parameters are used as standard welfare related behavior for stress indicators in the fish field. So it's maybe a good combination that we were possible to do this in this kind of studies, and it's also used in general in fish behavioral studies and welfare studies. Yeah, so we were possible to have video tracking of group house fish in different type of tanks. We were determine stress effects from the treatment, possible to determine the behaviors without human observation, and possible to have to detect early abnormal behaviors. In the carnivore groups, we tried to have something to do with ferrets, but the challenge was the different cages in every facility to have good options to track or to follow or to observe the ferrets. So there was one done in the Netherlands for group house ferret, but they were just held in one room with a video on top, so not in a cage. On some other facilities where ferrets are kept in cages. But we did a study in France with Sandrine Lessier with foxes, and there we couldn't measure the behavior automatically, or but we could use the observer to score the behaviors afterwards. So there were foxes kept and also implanted with temperature implants. So we had successful temperature implants to measure individually housed foxes, and we had good quality of the temperature results from that. And we had continuous recording of videos, and the behavior recognition couldn't be automatic. The analysis is still being done because it's quite time consuming to score the behaviors. But because the foxes were now observed for a longer period of time, new type of behaviors were found and observed. And you have fewer stressors for the wild animals because you don't have to come into the room, and also for the temperature measuring recordings that you could do that in the wild animals. In rodents, we only did trials with hamsters, and the challenge was for the rodents to have them individual data of group house animals. We were not able to do video tracking of the individual information of the group house animals, but we used an implant that also has activity measurements, that you could still define the individual activity of the group house animals. And those measurements were done with 3D accelerometer. And they were trialed in the rodents, different types of implants and capsules, but it all needs different surgical procedures. There was still support needed to interpret the data coming from the temperature measure, different temperature solutions. And from the activity and the temperature results, the hamsters, it wasn't possible to see clear clinical signs with normal observations, but with the two options, you could see really the effect of the infection on the animals. So conclusions on the design, standard models contribute to more comparable, reproducible outcomes in infectious disease studies. And rotating the monitoring equipment across the research facility, it worked, but it was quite cumbersome, laborsome, and time consuming. So it's easier to have a fixed option for the technologies. Acquisition, the automatic measurements for activity and body temperature in different species and individual at an individual level was possible, but real-time data was a question if it always is needed for the different studies. And the analysis, body temperature and behavior measurements are both useful across multiple agents and species to access and monitor infectious diseases, and continuous instrumented monitoring of behavior and physiological brings finding and insights that are easily missed based on the clinical observation. So you do see really different results than just going to a pen and look at the animals, because they do behave a bit differently. Then for the biocontainment implications for the equipment. So then, the advice is to have the equipment more installed permanently on facilities, and that reduces the need to reinstall the equipment. But the equipment that we all trialed in these studies performed better than we expected. Also, most of the equipment was possible to decontaminate, and there was minimal loss of the equipment in these studies. So what I heard from the previous packages was that you are building new buildings, or are working on it, or planning this. So then an advice is to have an option, at least for a camera, for continuous monitoring, that you can always see what happened inside the rooms, and also to have data and power connection possibilities inside the room for different equipment that you want to use, that is always an option. And the lessons learned and future developments. to standardize behavior and physiological monitoring for each animal model. The human endpoint detection, the thresholds still need to define. So first, for the group, it's to have what kind of behaviors do you want to have? What are the boundaries of the, when is it the behavioral point?
Caracteres. Bueno.
So the real, yeah. Yeah, so it is possible to have to define a human endpoint, but the agreement on what are the boundaries needs to be done. And what we want still to be able to have is an alerting function when behaviors are abnormal. An easy flexible solution for data acquisition across multiple rooms, because now you collect in one room each and then you have to combine the data. And remote access to the different data acquisition tools to monitoring the animals in real time. And yeah, the best is to have a software tool that can turn this raw data into useful information, because you get quite a lot of different piles of raw data exports of different solutions. So then I want to hand over the
okay, let's take the questions all together if we can. So Anna would be the next to present online.
Yeah. Yeah.
And Anna from FLI, who will comment on the models they are using.
Yes, good morning, everybody. It's so nice to see you all, at least online. Can I share my slides? Oh, yeah. This should be it, hopefully. Do you see the slides?
Yes.
Wonderful. Okay, so I would like to report a little bit on the experience that we made when we tested some of the equipment that Leslie just mentioned, and we tried it in different species. So yeah, we have quite some interesting experience. We worked with the body cap system in different species. That's here on the upper left. It's a small capsule that is either ingested or implanted subcutaneously, interperitoneally, wherever you want it. And this little monitor that you see, that needs to be in vicinity to the animal. So maximal is, I would say it's 2 meters. And if you have the animals in metal cages, it should be definitely less than 2 meters. So the metal doesn't make it easier, put it that way. On the upper right, you see the surgery room. It's a very simple surgery room, but that's where we implanted the TSE telemetry system. And on the bottom shows the StarLoggy system or data loggers that we have used in several, excuse me, several studies now. So start with the body cap system. We tried this in pigs first. So we wanted to know if this can be applied in pigs in an easy way. First, we put it subcutaneously in the area of the knee or medial femur area, because obviously we wanted to avoid doing a real surgery and implant it intraperitoneally. The operation was okay, and we had these animals that were piglets, 20 to 30 kilograms. We had them in a group of 4 animals, and that didn't gnaw on each other's wounds, so that was all no problem at all. But we did this in January in an area that is more or less outside temperature, and the animals nicely put the straw to the side and lied down on the cold floor. And that's why sometimes we measured temperatures of around 33 degrees. So this is certainly not the way to go if you don't have the animals in a warm place, which in challenge experiments usually they will be. But this needs to be taken into account. But apart from that, it worked well. Next, we tried, because of this experience, here you can, you see the fluctuation again and how low it gets. Oops, sorry. Next, we tried intraperitoneal implantation because we thought then the capsule is very safe and nicely warm. That also worked well. We had no complications during the operation, and this is 2 weeks after the implantation. So also no inflammatory reaction around the capsule. But already these 20 to 30 kilogram piglets were too large for data transmission. So these green points belong to the subcutaneous pig or subcutaneous capsule in a pig. So that's not much better than doing a rectal temperature daily. So that unfortunately didn't help at all. So conclusion from our side for pigs, if we want to use this system, it would need to be implanted subcutaneously in a warm area. Next, we worked with bats with the same system. We keep food bats here at the FLI, and we wanted to learn more about the physiological temperature of these animals before we do challenge experiments. And we did, again, in parallel, or in this case, we did it in parallel, subcutaneous and intraperitoneal implantation. The green dots that you see here belong to an animal where it was implanted subcutaneously, so the measurements worked well. Of course, the temperature is a little bit lower than intraperitoneal. That was to be expected. We saw here for the first time that the temperature really fluctuates across at least 5 degrees centigrade, depending on what the animal is doing. But the transponder was a little bit too large for the animals, so the skin above the transponder got thinner and thinner. So after 3 days, we had to take them out again because it would have ruptured probably the next day. So in this system, it worked technically, but it can't be done. But the intraperitoneal implantation, that's here now the yellow dots, worked very well. As I said, we see this huge fluctuation. The animals tolerated it very well. The operation went well and they recovered nicely. But this opens the question, how do we want to notice fever in a bat? So taking 1 rectal temperature per day doesn't make any sense at all. But we learned a lot about the physiology of these animals. So this is a summary of the temperature data that we collected. So this is the day, so 6 o'clock in the morning, the lights go on, the bats go to sleep. And 6 o'clock at night, lights go off and the bats get active, and the temperature goes distinctly up. We used this system then in an experiment that was funded by the Transnational Access from VetBioNet. Ghazi Kayali had applied for this, and we did an influenza challenge together with Donata Hoffmann and Nico Halve here at the FLI. And here you see the data. We didn't observe any clear differences in temperature, but we were able to monitor that we didn't see a difference. But that was also a very important result. So a practical application of this system in a challenge experiment. So my conclusion for the body carb system, it is easy to implant. It is well tolerated. By us, it was in all cases well tolerated, except for the subcutaneous problem in the bats. Data transfer is a little bit tricky. So that's why I have this picture here of the metal cage where we keep the bats during challenge experiments. So here we had to mount the monitor directly on the guitar mesh of the front door of the cage. So the signal wouldn't be able to transmit through a closed metal wall. We had some problem with the monitor. It was sensitive to the disinfection. We didn't dunk it, of course, but even wiping and spraying killed one of the monitors. And we were quite happy with the technical support. So whenever something went wrong, we got some support from them. So the next system we tested, oh, sorry, that's just a little excursion. You can also, with very simple methods, get some very important data. And here we just mounted a wild camera in the bat colony room. Again, 6 o'clock at night, lights go off, bats get active, and at 6 o'clock in the morning, lights go on and bats go to sleep. So we see nicely just from the frequency of the camera activation how we can monitor this activity. And if we put this into a graph, it can be summarized nicely like this. And if we put the temperature data together with these, you see a very good correlation of activity and temperature. And these data were acquired, honestly, with quite simple effort. So it can be done. In some cases, it's not always possible to do it with very simple methods, but sometimes it does. So next we started working with a telemetry system supplied by TSE Systems. This is a much more sophisticated system. You have these the transponder and leads to monitor the thoracic pressure that can then calculate the respiratory rate. You have leads to measure the ECG, and of course the body temperature is also measured. So this is a quite complex surgery. You need training for that. You need a surgery room. I mean, you see it can be quite simple, but you need a dedicated room that's clean enough to do this quite complicated and operation that takes some time. But if it's done, you have the real-time measurement and you can keep the animals in groups. So this shows it again, the principle of the system. You have this monitor and then the data are transferred to a computer. But to be able to do that, Melanie and myself went to a training in the Netherlands, in Almere, and that went for a whole week. It was very good. It helped me for all the other surgeries that we did afterwards with the telemetry, but it is a sophisticated system. So we tried this on one rat, and that really worked well. We monitored the data for several months without a problem. So I would say the system works well, but it's complex. Technology worked fine. We had no problems, and the support was also satisfactory. And we would continue with this if not StarODI crossed our ways, because now we can almost generate the same data with much less effort. And that's why we are using this system mainly right now. The only drawback of this, these are data loggers. It's not a real-time data transmission, but you can only at the end of the experiment read the data that were collected during over the time. They offer different systems. One of it just measures the body temperature. You can measure body temperature and heart rate, or body temperature and locomotion activity. And these are the loggers that we are using. They are not small, I must admit. They are borderline for hamsters, but luckily hamsters have a very loose skin, so there's enough room to place these transponders or data loggers. And this is what we did. So as everybody else, we did quite a few hamster challenge studies with SARS-CoV-2, and we applied these data loggers subcutaneously between the shoulder blades, and it worked quite well. The StarODI, they recommend to tie the data logger to the subcutaneous skin. So this is what's shown here. There's a little hole where you can put where you can suture it to the skin. In our experience, this is more of an irritation than if you just put it under the skin, and it will start wandering in the animal, but the animals didn't show any distress by that. And this is just an example. So we did a drug testing experiment. So this is the uninfected group. The red arrow always shows the time of challenge. And here the pattern doesn't really change. The red color is the body temperature. Blue is the heart rate that's calculated from the locomotion activity. and green is the locomotion activity. And you see nice regular patterns. Next to it, on the right-hand side, this is the animals that were infected. And you see 2 days after infection, the whole metabolism slows down in the animal. The temperature goes down, heart rate goes down, and the activity clearly also goes down. And here are 2 examples for drugs. One worked not so well on the bottom left, so it almost looks like the untreated animals. And the other candidate worked much better. It resembles at least the uninfected animals. So these data are very nice additive to the virological data that we are collecting during the studies. And another example, then we applied it also to fruit bats that were infected. Again, uninfected, you see this nice fluctuation. Here we only show temperature and locomotion activity. And then we used, excuse me, different inoculation routes. We didn't see any clinical sign in any of the animals, and we had almost no virological data that we could use to understand what's happening in the bats. But what struck us that the orally infected bats showed the most or the highest change in the patterns of temperature and locomotion activity. And from all the virological data, we would have said in the orally infected bats, nothing happened at all. The virus disappeared and they were just clean. So these data really gave us added information that helped us, or that we still need to figure out what exactly happened in the bats, but we know that something happened because they really reacted. Again, this is what the hamster looks like. So for Star RDE, I would say for now, in our hands, this is the most convenient and most practical system. It's easy to implant, the technology worked well, the support is excellent. Askia helped us very much also with the data interpretation because they were also interested how their system works. And as I said, the generated data are very important. Another little excursion, this is not telemetry, but this is also something that we tried to just understand better what happens in the infected animals. This was done in cooperation with Alex Rieck from the FLI in Celle, and they, or this method is applied quite regularly in wildlife to monitor the energy expenditure by injecting this doubly labeled water into the animal. And then after certain time points after this injection, you take blood samples, and from the blood, the water and energy expenditure can be recalculated. And we did that also in SARS-CoV-2 infected animals, the same experiment that I showed before. So different infection routes versus non-infected bats. And here we didn't really see a difference. So obviously also we didn't see any clinical signs in the bats. So they were largely unimpressed by the infection. But to show that the system itself works well, this is how it looks in SARS-CoV-2 infected hamsters. So on the left is an uninfected group. In the middle is an infected and treated group. And here on the right is the infected group without any treatment. And they really lost their appetite, obviously. So the system works well. And in conclusion, so the telemetry options that we have now really offer us great opportunities to get additional data that are, depending on the research question, can be really important and give us very important information. Leslie has also mentioned that the regular monitoring of the animal gets more and more of a requirement by the animal welfare authorities. Then that's mostly video monitoring, but also other data could add to that. You can have non-invasive possibilities to detect some changes that we would not have noticed without this. But of course, it needs to be adjusted to the certain research question that you want to answer. This is definitely more effort than doing a challenge experiment without this. So you really need to decide, do I need this data? Do I expect any additional data that will help me interpret? Or is this just a nice toy, creating nice graphs, but saying the same that you would have answered without this? So I would say we really need to go further with this and dedicate it more, and the systems develop further also. And I would, of course, like to acknowledge all the colleagues that were involved in this, quite a few people at the FLI with all the different systems that we tested and surgeries that we did. Gazika Ayali was the colleague that supplied us with the influenza strain that was then tested in the TNA experiment, and the 3 manufacturers of the telemetry systems. Thank you for your attention. I don't hear anything.
Jim is present.
What have you got? You haven't downloaded.
Yeah, we can. Dat we switchen aan
Show it after.
Yeah.
Do we switch or what?
Yeah.
Switch order.
Change order.
Francesco Pascoli.
Yeah. On fish.
Yeah. Yes, on fish. Shall we go to fish?
Hi. Okay, so. Okay. Okay. Hi, everyone. Very quick because we are late, so I try to make it as quickly as possible. So we were asked to perform a pilot study on behavioral analysis using the video recording, and so the setup of the trial was using 2 tanks of 300 liters each. One was we had the fishes infected with the nodovirus, that is the virus already explained by Lucia on the first day during TNA, so I skip it. And the other one was just mock infected, so control fishes. The setup was with the 2 cameras over the 2 tanks, and we need to add external lights to have a better view from the cameras. The first initial critical points was to make everything waterproof, because as you know, salt water and electronics devices is not very friendly. And so we need also to stop the water flow and the air inlet into the water, because it was disturbing the recording and the subsequent analysis. So the problem is that in water, the oxygen decreases very quickly. So this allowed us to make a maximum of 30 minutes of recording per time. And the other thing is that fish, in this case, it was sea bass, are crepuscular animals, so they don't like high looks in the environment. So the putting the external light was quite stressful for them. Anyway, we record 30 minutes of videos per day, 2 times per day, one in the morning, one in the afternoon, and we perform the recording 10 days out of 21 in total of the observation. And we after that we analyzed the data with the Etovision XT from Noldus Technologies, and we used these parameters for analysis. So it's velocity of the fishes, the distance, the frequency of moving, and the shoal activity, that is the group behavior. And it was impossible to track single animals, so the analyses were made by group. And I just show you very quick, just to have an idea on what is different. This was these are infected fish that are randomly moving into the tank, whereas the mock infected stay more in group, and the velocity is higher than, as you, these are real time. So yeah, you can see also by without analysis, I mean, the difference. And the same, it was just to highlight a little bit. This fish is swimming belly up. That is a typical clinical sign of the betanodovirus infection. And as you see in this moment, this disappear from the cameras because of the similar pattern of the bottom of the tank. So this was a problem because this animal doesn't give doesn't give any. So very quickly on the results. In general, infected fishes were lower, were slow, and like they swim very very low, and the most important data is that the shoal activity is very high in the mock infected group and very low in the infected one. And so by conclusion, we need to improve the optimization because yes, the belly up is a human endpoint, for example, in this situation. So we need, it could be a very interesting point to highlight by automatic recording. The disease, of course, influenced the behavior. So the movement is reduced. Is very interesting for a possible forecasting prevision. So to predict what is happening in a tank, and of course also in the welfare. Leslie mentioned before, for welfare studies, it could be very interesting because you can observe the animals without the human interactions, of course, and record all the behavior that could be interesting for in this case, enrichment studies, because it's very in fishes is a very challenge and very discussion made by the government on what is the enrichment and what is the importance of the enrichment inside the tanks. So that's it. 3.
Yes.
All right. Good morning, everybody. I don't even have a title slide, so to save time. So I'm Tim Connold from APHA. Right, we tried something different. It's infrared thermography. For those who don't know what that is, just some background information. So infrared radiation is emitted from all objects at temperatures above absolute zero. So if an object is warmer, it emits more infrared radiation. You can't see it. It's a method where you can, if you use infrared thermography, it's a method to detect the radiation. And you can use cameras, and there are different models here. So we had the one on the left. There's also some sort of iPad type camera that you can purchase. And then on the right, that's called a spot pyrometer. So that doesn't convert it into a visual pictures. It just measures the temperature of a spot. So if you hold it against an animal, it detects the temperature, and it's just a reading basically of a temperature. With these cameras, you get actually a picture. And if you then basically get radiation that you detect, so that it's not just the radiation from the target object, so it's also from its surroundings, and that could be a problem. So you have to adjust for that, because there could be basically humidity, the outside temperature, that all affects the reading, so that you can adjust it usually with software. Oh, that does, oh, here you go. What you have to keep in mind when you use it in animals is animals can certainly mammals can regulate their core temperature, and they can, for example, use vasodilation or constriction. So basically, if it's too hot, then they will vasodilate so that they emit more heat, and equally, if it's really, really cold, they constrict their blood vessels, and that has an effect obviously on the radiation you will detect. There's also a change of insulation. You can, for example, in mice, you get piloerection to keep them basically warm. Then you've got, or coming colder, some animals might sweat or hyperventilate to get rid of heat. So there's a lot of measures to affect or to regulate your core temperature. And that's why sometimes measurement of the skin temperature might necessarily not work with infrared thermography. Then you also have to keep in mind that animals are not stationary, they move about. So that's why single pictures are probably less useful than having video footage. And that's the reason why the camera we purchased had actually the option to record videos. And then the other problem is that you've got all these hair, fur, wool, feathers that influence reading, but basically you can't read anything. It just is one blur on the camera. So you have to either find areas where there's no insulation, or you just shave the area off, basically what you want to monitor. And if you want to do it under uncontrolled environment conditions, you have got then problems with the temperature, the outside temperature, humidity, wind, and all those things that might affect it. So it's better to use it in a high containment facility where you've got basically controlled environmental conditions. And then of course the other thing is your camera needs to withstand disinfection, fumigation. So we still use formalin to disinfect. You can also use ethanol. We tried both fumigation and wiping with ethanol. So we did 3 studies here. One is highly pathogenic avian influenza. So if you like hypothesis, that's all a hypothesis for each individual study. So we want to know if you can maybe refine an experiment by measuring temperature in highly pathogenic avian influenza affected chickens, because we often had the problem that they die suddenly. Then for African swine fever, we heard that was in the first day. So there was a study with African swine fever was in wild boar. We were hoping that we were at the time we were concerned that we might not be able to take regular temperatures, and we thought, well, if you can use infrared thermography in addition, or then we might spot fever. Well, it turned out that actually domestic pigs were worse in taking regular temperature than wild boar. And then lastly, the bovine tuberculosis. You see the skin tests. If you wonder why there are 3 shaved areas, that's because we tested another reagent. Currently do a DIVA test study, so it's usually just 2, and then you measure basically the skin thickness, and we thought maybe you can determine a difference basically after the skin test injection, that you can see temperature changes and that might be different from non-infected animals. So first of all, you just hear the results. Yeah, that's fine. Okay, so what you just see is we had some survivors. We didn't have any negative controls as such, but we had survivors. But these are our negative, well, uninfected, well, not infected, but recovered controls. And you can see there's a wider amplitude in those ones that are infected and did not survive. For the African swine fever, you have on the bottom the graph, and these are on the lower temperature graph. These are the temperatures of the eye. We use the eye as a comparison also because we thought maybe that represents more the core temperature. So the temperature is lower. We've got then the area next to the eye, and we also have rectal temperature. It's very difficult to see, but it seems to correlate quite nicely. Rectal temperature slightly higher than the area next to the eye, but you can still see that it more or less correlates quite well, and it rises basically as animals got sick and become febrile. And then lastly, that's for the TB reactors. So if you look at the dots here, so that's mean and standard error of mean, you can see that animals with visible lesions have basically a higher temperature than those ones that have non-visible lesions when you do a postmortem, and the controls had even less of the temperature change. So that's the amplitude basically between the lowest and the highest temperature. So it doesn't take into account basically the time at the moment. Similar, so that you use normally two inoculations. So one is the avian tuberculin, the other one is the bovine tuberculin. So in both instances, you've got basically changes or differences between the one with visible lesions and non-visible lesions on controls. So what we conclude is that you can use infrared thermography in high containment. We haven't done all the data analysis, but it looks quite promising. So you've got an excessive rise or drop in the head temperature towards the end stage in avian influenza. You've got also a rise in rectal temperature in wild boar, and that's usually accompanied by a rise in the skin temperature. And if you look at the TB reactors, so the mean injection site temperature amplitude is higher in those ones that had visible lesions. And the further work is complete the analysis and publish the results, and then collect more TB skin test reactor data. And we also want to look at the time over time, basically, if there's a difference. And that's more or less it. We acknowledge the people. That's always important that we use studies that are currently running at APHA, and then we use VetBioNet basically to make use of the studies to try our infrared thermography.
Thank you very much, Tim. We have to apologize. We were running out of time. So in fact, Wieneke, the Wageningen WBVR, they presented in the Brussels meeting a very nice thing. That won't be repeated now because it will be in your coffee break. Please approach Norbert or his colleagues if you want details about that. It is nice, but not fitting anymore in this session. What I liked about thermography is the psychedelic pictures of animals, beautiful. What we heard from everybody is that they saw things, time-bound things or location-bound things or behavior-bound things that inform them more about the internal state of the animals than the classical approaches. Secondly, that using all that kind of technology in high biocontainment is a challenge by itself. And I would like to thank everybody for their attention because the coffee gets cold.
Well, you know, from the previous days, when lunch is served, that takes some time also. So, and then there are people that have to leave a bit earlier, just as Anna now leaving back to Italy. So let's try to be let's try to stick to the schedule proposed. And so we will switch now to the second GRA session. That is work package 7, and I have the pleasure to pass the mic to Esther.
Thank you, Sasha, and thank you, everyone. Welcome to this session. Since we are on a hurry, I am just remind the main objectives of this work package is the number 7, is focused on the experimental models for animal infectious diseases. Yes, pass. So the main objective of this work package is the development of quality and ethical standards of research on episodic and zoonotic diseases by establishing novel or optimized infection models, live animals and also in vitro models. To do so, work package 7 has as a specific objectives distribute in 5 different tasks that are focused in the different animal models. The first one is for pathogenesis and host immunity studies. The second one is for development of reservoir host infection models. The third is related to airborne infections. The fourth is for aquatic animal infection models. And the number 5 is to develop alternative infection models based on immortalized cells, 2D and 3D cell cultures, be able to do explants, embryonic chicken eggs, and others. This work package have 5 milestones that were all finished and submitted, and 6 deliverables. 4 out of these 6 deliverables has been already submitted, and we have to submit 2 deliverables, one focused on immunity models, and the other one related to airborne transmission and susceptibility. These deliverables, or these documents, are under work and will be sent in this month for the end of the project. So we have organized this session, the work package 7, with some presentations from members that have participated in this work package. So we will have, the first is to thank all the speakers that are going to participate in this session. We have 2 members representing the task number 1. The first one is Rodrigo Guabiraba Brito from INRA. That is going to talk about mucosal immunity in the chicken. Second one is Daniel Rodriguez Martin from INEA. He is going to talk about peste de petit ruminant in sheep and goats. Julia Vergara from IRTA CRESA is representing the task number 2, and she's going to talk about pathogenesis of MERS coronaviruses in llamas and alpacas. Susanna Commander from BBVR Netherlands, who will be present results in host environment influences in co-infection in pigs. This presentation will be online. I think Susanna is already connected, so welcome, Susanna. After we have the lunch, and after lunch, we have 2 more presentations. The number 4, or task 4, that is aquatic model disease. We have Patricia Diaz Rosales from INEA, who is going to talk about rainbow trout pathogens and infection models. And 7.5 task, that is the in vitro models. We have also an online presentation from Tobias May from InScreen Germany, who is going to talk about immortalization strategies to establish novel animal in vitro models. The first presentation is Rodrigo, but first to pass to Rodrigo presentation, I want to congratulate to Julia Vergara, who has been awarded with the prize to the best young researcher in Catalonia. So congratulations. And now we can start with the session. So Rodrigo, please.
Morning, everyone. Let's see. Just make a quick test here. No. All right. Yeah, looks like working. So good morning, everyone. Thank you very much. It's a pleasure to be here. So today I will show you a bit of what we have been doing in the scope of the VetBioNet, especially on the work package 7, which is to uncover the core principles of the gut-lung axis to enhance mucosal immunity in the chicken. This work was essentially performed at the INRAE with our preferred partner, the PFU, as an experimental unit linked to the VetBioNet project as well. So just as an overview, a quick overview, we know very well right now that related to poultry and the goat microbiota, that this is very well interrelated. So well-being and performance in poultry is linked to good gut health and a balanced gut microbiota. So because this microbiota host balance ensures immunity, development of immunity, and resistance to pathogens. There are several problems right now in commercial production that people are starting to ask questions about, which is essentially in modern animal husbandry practices. There are thorough hygiene, hygiene practice and the absence of chick-hand contact especially, but also advanced selection and production traits with may lead or not to unknown physiological consequences. So all of this may have an impact on the early establishment of a well-balanced microbiota, so with potential negative consequences to immune competence in poultry in later life. This is because the gut microbiota and its metabolites are well known, especially in mammals, to regulate the local and systemic innate immune system and innate immune responses. Metabolites from the microbiota, such as short-chain fatty acids, are involved in myelopoiesis, leukocyte maturation function, not only in the bone marrow, but also in distant organ sites. And we know very well now in mammals that those extraintestinal effects of metabolites play essential roles in the lungs, which I will discuss a little bit today, but also in the brain, which led to coining terms such as the gut-lung axis or the gut-brain axis. So short-chain fatty acids are greatly involved in lungs mucosal immunity. That's what is highlighted here on the right very quickly, showing that the presence of this metabolites within the alveoli or in the parenchyma of the lung are involved in the control of inflammatory responses, the expression of antimicrobial peptides, and so on. So there is a role, a recognized role of these metabolites outside the gut. And that's why we talk about here the gut-lung axis. So this has started to be much more clear when changes in the composition of the gut microbiota in humans, especially what it calls dysbiosis, increases susceptibility to respiratory diseases that can be chronic disease or infectious diseases in humans. And other key studies showed that mice have an increased predisposition to allergy following early life antibiotic usage. But all of this, although more or less studied right now, are unexplored concepts in most farm animals, including poultry, even if common poultry diseases affect both the gut and the lungs. But we already have some evidence in poultry. For example, in ducks treated with antibiotics and infected with IHIPAF H5N9 strain, you have more virus excretion and decreased antiviral immune responses. In chickens, animals treated with antibiotics and infected with H9N2 influenza have higher excretion as well in both in the respiratory and in the intestinal tract. So there's a consensus that there is a role of the gut microbiota in the initiation of the innate immune response against influenza infection, but the mechanisms are largely yet to be defined. So that's where we are placed. our team, Trimo, which is led by Sasha Trapp. And we have two main questions. I will focus essentially on the first one, which is the role of the gut microbiota on the functional development of the mucosal immune system in the chicken, what we call the gut-lung axis, but also the participation of the gut-lung axis in antiviral immunity, notably in regard to influenza infection. And this is important in the context of VetBioNet because we've been using the germ-free models that were set up at the PFU, at the INRAE here in Nosy, in Tours, since 1969. So these models of chickens have evolved technologically. You have pictures here showing how do you raise conventionals and germ-free chicken in isolators and BSL2 levels. And this has improved a lot to the point that now we can have inbred chicken lines or commercial chicken lines that can become germ-free chickens in these structures. Then we can go ahead to perform studies on infection or characterization of the immune system. That's what we started to do, and the data will show you. So metabolomics, transcriptomes, and flow cytometry analysis normally follow this analysis. I will focus essentially on metabolomics and transcriptomics today using immune competent chickens, where no maternal antibodies are evidenced anymore, so they become immune competent. So these chickens grow very nicely. They have essentially no major physiological differences, but as we see, they have immunological differences. So one of the first things we started to do is, can we detect metabolites coming from the microbiota outside the gut? So we know, like looking at the cecal context, the microbiota enriched portion of the chicken in the ceca. So we have a lot of short-chain fatty acids, acetate, propionate, butyrate, and which is essentially absent in germ-free chickens. Acetate is essentially coming from the feed, but it's essentially absent in germ-free chickens. But what is interesting is that what I highlight here is the presence of short-chain fatty acids in the lungs, which are in low concentrations, but can be detected anyway, which are largely absent in germ-free chickens in blue here as well. We did the same in the spleen, we did the same in the blood, so they are reduced or absent in these other compartments as well. So local metabolism can produce these metabolites, yes, but to an extent that is undefined or absent for certain organs, like the lungs, for example. No evidence that butyrate can be produced in detectable amounts in the lungs. We went ahead going into the organs. I will try to do it quickly as well. It's just an overview, but looking at the ceca of germ-free chickens, the transcriptomic signature highlights that cell cycle and metabolism are drastically altered in the ceca of germ-free chickens, especially with dramatic changes in epithelial cell physiology related essentially to cell division and so on, chromosome stability, etc. And looking at certain genes related to mucosal immunity, and there are dramatic changes as well. Certain are heavily downregulated, especially IL-22 receptor is a soluble receptor regulating IL-22, which is really important to mucosal immunity. Matrix metalloproteinase 1, which is essential to tissue repair and the stabilization of the intestinal tissue, and also several interferon stimulated genes such as OSL, EFIT5, MX1, EFIT-M3, and mucosal immunity like the IL-22 or IL-20 family. So this could be expected, and this is clearly seen when we do a PLS-DA analysis, where we see that germ-free and conventional animals are drastically different. So looking at the metabolic parameters, we have especially for certain amino acids, we have alanine, aspartate, and glycine. We have a strong reduction in amino acids, and that's like a metaboanalyst analysis shows clear that several pathways are heavily altered, most of them related to the TCA cycle, which is a bit obvious as well, but that pinpoints to a regulatory role and a connection potentially between the TCA cycle and the regulation of the local immune system, which is known from mouse and other mammalian species. Then we took a look at the lungs, and although we have a lot less differently expressed genes, we have some, and most of them related to the immune system. A couple of apoptosis, chemotaxis, cytokines, on things related to ontology for these genes. and we take a closer look, we observe that genes related to phagocyte function and toll-like receptors, cytokines, are drastically altered, upregulated or downregulated, but they are different regulated in germ-free chickens. So at least mononuclear phagocytes, or the mononuclear phagocyte system, is altered in these animals, but not only. I'll show you later. And together with the metabolism, the metabolomics analysis we did, we observed, for example, important difference in glutamine and glutamate metabolism, which is linked to biosynthesis of nucleic acids and proteins. So the metabolism is okay, apparently, but there are things that are different. Although the relationship between the metabolism and the innate immunity we are observing are not yet that clear to us. And the PLSDA, to try to push a little bit further, reveals as well that ISG and interferon stimulated genes are also heavily downregulated in the lungs. And then we choose the spleen as the main lymphoid organ in the interface in the middle of the chicken, let's say, in the pathway gut-lung axis, and we observe a couple of alterations more than in the lungs, things related to metabolism, to immune system, especially the complement, which is quite interesting. With things such as OSL again and ISG, upregulation of avian beta defensins. That's very curious. We could not explore that much yet, but there are interesting information going there. But the general message is that the immune system, some key things are downregulated. And the metabolism goes hand in hand with the other things we saw in the other organs, especially here, for example, in the ammonia recycling or the glycine serine metabolism, all of them involved in the TCA cycle again. So overall, these animals have an alteration of the metabolism along the gut-lung axis. So this is just a Venn diagram select from a couple of analyses we performed, and taking the most different expressed genes from the cecal, lungs, and spleen, we get a couple of genes that are common to these organs, and most of them, except for the GLUL receptor, for the glucose metabolism, all the other genes are essentially ISGs. So they are interferon stimulating genes, which clearly highlights that these things can be interesting in the context of viral infections. And this is just an overhaul, like a conclusion of what we saw, which is essentially pretty much what we see in mammals as well. There is this concentration gradient where we have more acetate and less butyrate and coming from the gut and going to the periphery. This was observed already in humans and in pig, if I'm not wrong. But there are essential other differences. For example, where do these metabolites will act? Because in the chickens, some receptors are absent, and some others, for example, FFR2 could recognize butyrate, but other metabolites, we have more than 20 paralogs for this receptor in chickens, which is, people believe, is related to domestication. So how chickens deal with short-chain fatty acids physiologically is not clear yet, which complicates things a lot. And then to this final part, what we try to do is, since we have all these alterations, could we have try to select one of these molecules and take a cell line, for example, in the lungs, if you're interested in the gut-lung axis, and for the absence of a good chicken intestinal cell line, which is the case right up to now, we choose our cell lines, which the CLAC213, which were cells developed in our lab a couple of years ago. They are unique cells representing the respiratory epithelium of chickens. What we did, we tested all short-chain fatty acids, and butyrate gave us the best response. They respond to butyrate in different ways. And what we observed, we have differences in metabolism, alanine metabolism, urea cycle, nothing outstanding, alterates a bit, but not that strong, which is good if you are looking at gene regulation, for example. And that's what we did. We took a look at inflammatory genes. They are a bit upregulated in the presence of butyrate, but not that much. ISGs like MX1 strongly with antiviral effects, not touched at all. But then we look at OSL, the same gene we saw for several others of our analysis. And then this gene is consistently upregulated in a dose concentration manner, time-dependent manner. It's interesting because OASL, different to OAS1, which is a lot very studied in mammals, has a role that is undefined to not that clear even today, which is essentially working through degradation of double-stranded RNA in link with RNase L to provide RNA to reguide, for example. So this link that could be direct, being antiviral, or giving support to reguide functions and being antiviral. But the point is, there are no reguide in chickens. Chickens have no reguide. And this is another TNA project, by the way, with Ishan Seed and Benjamin Schusser. But the point is, we are curious about this molecule because there are no reguide chickens, and there are strong correlations with ubiquitin motifs that are found in OSL. They are linked to ISG15, which is another ISG, which is absent in chickens as well. So would OSL play a role in that? Would OSL be the ISG15 in chickens? So we are currently working on this regulatory mechanism, but it's clear for us that butyrate regulates OSL in an interesting fashion, which could explain how short-chain fatty acids may regulate ISGs in peripheral organs. So just to assess a little bit if butyrate would have viral effects, antiviral effects, sorry, we infected our SCLAC models with an H1N1 low-path strain. We work in our lab. We consistently observed a mild reduction in viral load. It's consistent, but it's not even one log, but it's there, which is correlated with an upregulation of OASL in time. This is just one time point, but if you have the virus and the butyrate, we have more OASL than we would expect. We are currently doing and silencing OASL to see its role in viral infection and try to establish other parameters. We're also working on which transcription factors are involved in the regulation of OSL. And what we found right now is that it's a transcription factor that is known to be regulated by butyrate in other species, which is SP1. But SP1 regulates, it have binding sites in OSL promoter, which is not the case for humans and pigs, for example. The presence of SP1 binding sites in OSL promoter is only visible, is only clear in birds. So another thing that could be explored, and it's very particular to the species. So to summarize, the chicken microbiota seems essential to immune system transcriptional regulation, which is from the intestine to the lungs. We could detect short-chain fatty acids along the gut-lung axis. So the gut microbiota is always involved in regulation of metabolic pathways, TCA cycle, amino acids, and genes encoding for ISGs and mucosal immunity molecules, although we cannot yet establish a clear relationship between metabolism and transcriptional regulation, which we expect to do with more accumulated data within the next months. So we believe the gut-lung axis revealed or partially revealed in the chicken with this intertwined dialogue between gut microbiota and the respiratory tract being large and explored. We think with the germ-free model, we could use it, and at least with showing dramatic difference, we can start to think about how to inform, optimize the feeding strategies or novel probiotics or postbiotics applications that could improve poultry resilience to pathogens affecting the respiratory and the gastrointestinal tracts via the regulation or get back to a good regulation of all the genes and metabolic pathways I just showed you before. So thank you very much. I especially thanks my lab and the people from the PFU, which are essential for the production of these animals and all the support they give us. So thank you very much. Not working. Come on, please. Thank you.
Why did you focus on OSL rather than on IRF1, which is very well established?
Yeah, I know. Because it was essentially because it was very, even in chickens, IRF1 established. Or,
is one of the genes that was in your better
I know. Yeah.
Everywhere.
I know. I know. It was most because, I mean, it was a choice to show this today because you also see TGF beta moving. But the TGF beta thing, which is well known in mammals, The whole story with SP1 and so on is absent in chickens. The promoter is not, there are no binding sites in the promoter. And the IRF1 regulation is chickens is unresolved. It's not very clear. We don't know well what is it doing clearly. We can identify that some diseases and so on. It's worth doing. Yeah, we plan to check for selected genes. We bet on the OSL because of this particularity of being absent, the promoter thing with SP1, butyrate acting there. And it's very interesting. I think for people working on virology, it's still worth getting a bit further on it because all the other ISGs do not move with butyrate. We didn't understand that. We were expecting so IRF1 could move with butyrate if we add to the CLEC cell line. It doesn't happen. Just a couple of cytokines. OSL, the only ISG. So we were curious to see.
So Martin has a question.
Yeah, thank you for this very nice presentation.
Go ahead. Otherwise, the online participants cannot hear you. Yeah.
Very nice presentation and interesting how microflora interacts with the immune system and also in responses to pathogens. For practical purposes, do you think that poultry industry could benefit from inoculating the newly machine hatched chickens with a certain defined microflora?
I believe yes, totally. And I think that's the strategy that is trying to be applied. Some people are proposing, for example, in ovo, like put in the egg, outside the egg. For example, what we call a maternal flora. By the way, we have a project with an industrial partner on this topic, especially related to the gut-lung axis, where we propose maternal flora or probiotics to see if we can see these changes from the gut to the lungs in face of influenza challenge. I didn't show the influenza challenge, we had no time, but maybe in the next opportunity, of course. Sasha's probably frustrated, but I can deal with it later.
We have a question, Paul, in the web.
Sure.
Ask a question.
Hey, does my audio work?
Yes, yes.
Great. Thank you for the nice talk. I was impressed by this germ-free model, and I'm curious to know if it's germ-free over the course of the entire experiment. And there's a question related to that as well. But first, this one.
Okay, so if they are germ-free all along, right?
Yes.
They are. We do routine tests.
All right.
Like routine tests every 4 or 5 days, something like this.
Right.
And we can keep them germ-free for a long period of time.
And in order to ensure that they're germ-free, do you apply antibiotics during the experiment or not?
To make sure they are germ-free, you mean?
Yeah. Yes.
No, we try not to introduce antibiotics at all.
Right.
That's the point. I mean, we
Yeah.
try to create this empty vessel the most naturally we can. And some people asked us before, because I mean, some things were identified, like the microbiota coming from the hand through the oviduct and things like this. I saw a nice publication with this. But the point is, after disinfection and the whole procedure we do, which is published, we published the procedure on Jovi. The animals are completely germ-free. So if there is a maternal microbiota thing going to the egg, by the time we disinfect and they hatch in isolators, this microbiota is not there anymore.
Oh, very impressive. Thank you.
Okay, thank you. The model is clean overall.
Thank you, Rodrigo. We pass to the next presentation from Daniel Rodriguez Martin from INEA, who is talking about pest ruminant.
Good morning. Thank you very much. As Esther said, I'm Daniel Rodriguez Martin. I work. Oh, do I have the thing? I think I need this. I need this. Thank you. I work at CISA in Madrid. Now today I'll show you part of the results we got for task force 7.1 in our lab, which I titled Pest epidemic virus infection disrupts dendritic cell responses to suppress immunity. Pest epidemic is an weakening and acute disease that must be identified to the World Organization of Animal Health. Its main hosts are goats and sheep, although goats are more sensitive to the disease. Transmission happens by direct contact between animals through secretions and aerosol. And PPR leads to leukopenia immunosuppression that opens a window of opportunistic infections for like pneumonia. Morbidity and mortality can reach up to 100% in vulnerable animals, and its etiological agent is Pest epidemic virus, or PPRV, which is a Paramyxoviridae family genus Morphellovirus. In this group, there are other relevant viruses like measles virus, canine distemper virus, and rinderpest virus. This last one already eradicated more than 10 years ago. This is a distribution map of the disease. In green are the areas officially free of PPR, and in gray those countries that cannot claim that status. As you can see, PPR is spread across large areas of Africa, Middle East, and Asia, with terrible economic and social consequences for a great deal of communities that heavily depend on sheep and goat as their main livelihood. The European Union is surrounded by the disease, so we are at high risk for the disease to cross our borders, and that's why it's so important to learn more about PPR and its causative agent. PPRV infection causes immunosuppression, although the mechanisms are not fully understood for the moment. We know from previous work in our lab that PPRV impairs T-cell responses in sheep, as you can see in the study spot result, where sheep infected after 7 days, the number of cells able to produce interferon were significantly reduced compared to naive sheep, sorry. We assessed the effect of PPRV infection in dendritic cell functionality, both in sheep and goat. And our goal was to find out more about the mechanisms behind this immunosuppression and to learn more about the different susceptibilities between goats and sheep. And to do so, we use dendritic cells that we got from differentiation or by differentiation from monocytes. I'm gonna talk now about the results we got with sheep, and later on at the end of my talk, I'll mention a few preliminary results with goats. We first checked the ability of PPRV to infect dendritic cells, and to do so, we use an antibody against PPRV-N protein attached to a fluorochrome. As you can see in these flow cytometry results, those PPRV-infected cultures, the fluorescent levels were higher in dark green for PPRV-infected cultures than mock-infected, meaning that PPRV could infect dendritic cells. And moreover, plaque assays showed that this infection was productive. We performed a transcriptomic analysis of both culture dendritic cells, cultures infected with PPRV and mock-infected. And as you can see in this principal coordinate analysis, the infection changes the expression of patterns, the expression patterns of dendritic cells infected with the virus. This volcano plot on the right-hand side of the slide shows that almost 700 genes were upregulated or downregulated when the virus infected dendritic cells. RNA-seq analysis of this result show that PPRV could change several signaling pathways. As you can see, the virus could change, or when dendritic cells were infected with the virus, there was an upregulation of several genes related to viral recognitions, meaning that dendritic cells could detect the presence of the virus of this infection. However, the several genes related with interferon were downregulated, while other genes related with TGF-beta were upregulated, meaning that the virus could interfere in the classical response the dendritic cells would give to an infection. So we wanted to learn about the changes in functionality of dendritic cells when infected with PPRV. We first assay the expression of certain key cell markers for the dendritic cell functionality. And as you can see, markers such as important as MHC1, MHC2, and costimulatory molecules CD80 and CD6 were then regulated in PPRV infected cultures in dark green, comparing to mock infected cultures in bright green, meaning that PPRV could, in a way, alter the ability of dendritic cells to mature. We performed as well an allogenic mixed lymphocyte reaction assay, or MLR, as dendritic cells, one of its abilities, when activated, when mature, is to activate T cells and make them proliferate. As you can see in these results, those T cells that were co-cultured with dendritic cells previously infected with PPRV in dark green were not able to proliferate, while those T cells co-cultured with dendritic cells mock infected in bright green could proliferate. This would mean, again, that PPRV could make dendritic cells or make them not to be able to have the ability to activate T cell, which is, by the way, key for a proper response. When we went back to our RNA-seq results, we checked some signature genes related to tolerogenic profile activity in dendritic cells, and we found out that a lot of them, the majority of them, were upregulated, including IL-10, which is a key gene for T cell activation, or INHB1, which is as well very important in the activation of TGF-beta. This could made us think that maybe PPRV wasn't avoiding maturation in the dendritic cells, but changing its maturation profile from an immunogenic one to a tolerogenic profile. And to check out this hypothesis, we performed an autologous T cell proliferation assay. And as you can see in these results, those T cells after infection, those CD4 and CD8 T cells that were co-cultured with dendritic cells previously infected with PPR has a lower ability to proliferate comparing to those T cells that were co-cultured with dendritic cells that were mock infected after concanavalin stimulation. So this would mean that, as we were suspecting from previous data from RNA-seq, that PPRV was swift in changing this profile, activation profile, from an immunogenic profile into a tolerogenic profile in dendritic cells. Now I'm going to talk about some preliminary results we got from goats. We check as well the ability of PPRV to infect dendritic cells in goats. And as you can see in these flow cytometry results, you can see that those PPRV infected show more signals. So it means the dendritic cells got infected. But, and here's the difference, this infection doesn't seem to be productive. We check as well some key cell markers in goats, and we have to point out some results. Basically, CD80, CD86, and CD40 expression was upregulated in PPRV infected cultures. And when it comes to allogenic T cell proliferation assay, we've got results quite similar to the ones we got for sheep. Those T cells that were cultured with PPRV infected dendritic cells could barely proliferate in gray, while those T cells that were cultured with mock infected dendritic cells, however, could manage to make T cells to proliferate, meaning that this mechanism is not what's making the difference between sheep and goat when it comes to susceptibility, so as I said, goats are more susceptible to PPRV than sheep. So summing up, PPRV infection induces changes in sheep dendritic cells transcriptome, modulating signaling pathways involved with viral recognition and cytokines. PPRV interferes in the differentiation and maturation process of sheep goat dendritic cells derived from monocytes, altering cell marker expression and T-cell stimulation. And PPRV infection promoted a DC activation profile that favored the induction of tolerance instead of the activation of antiviral immune response in sheep and goat. And that's it. Thank you very much.
Thank you, Daniel. Just an additional data about the results that you have shown that are related to the main objective of Work Package 7 is to mention that they have tested the susceptibility in different breeds of sheep. And they have found that the Churra breed, that is a Spanish sheep breed, is more susceptible to the PPRV infection than, for example, the Merino ones. So I think that this is an relevant information for the optimization of infection models. That is the main aim of this work package. So quite interesting, Daniel. So any question?
When you infect your sheep dendritic cells with a high dose,
yeah. Oops, I think, should I?
Is the virus killing the cells or not?
No, they're not. Actually, I didn't show because I don't have the time, but we checked the ability of, well, how they got in apoptosis, early or late apoptosis. And the virus is not, it's avoiding cells to get in apoptosis, in the apoptosis process, and they keep being alive for more than expected in vitro. So the virus seems to be not interested in killing the dendritic cells, but just to control them.
Because if you check the data, I saw the plot, you have, you make the virus, then goes down and then goes up. Okay.
But that's
never got upper than your inoculum. You know?
That's when you put, well, that's more, I think it's more like a technical thing. When you put, what I put there is like the input, what it's left of you put in the virus in the plaques, and this go up and down. That's because all of, I think,
get inside of cells.
Yeah, precisely. And then afterwards it goes up, and you get the virus in the medium, and it goes up for sheep. But for goats, surprisingly, they got infected, but you can tell that they are not, it's not a productive infection, and it's something different. Maybe could be part of the reasons why goats are more susceptible in a way from sheep. I don't know how, but seems to be like the opposite, but I don't know.
And did you check the cross presentation, for example, if you infect a cell type where the virus is actively replicating and you show these cells to your dendritic cell, what's happened? Because maybe
like the other way around, like I'm putting infected, let's say T cells, B cell, whatever. And no, I didn't do it, but it would be nice to do it. Yeah.
Thank you very much.
Thank you.
More questions? No? So in the web note, thank you.
I just want to say I'm so sorry to rush everyone, but we have to make efforts that at 12:50 the latest that we have lunch. Then we are already half an hour late because people must leave and they want to eat a bite before leaving also, okay? And would be nice also if we have just 1 minute time after to take a picture all together. Okay.
So we pass to Julia Vergara, is the next presentation.
Thank you very much. Yeah, I'm sorry, Sasha, because you asked me to present after lunch, but we need to leave. So I'm not sure if you are expecting me to present much data about immunopathogenesis after MERS infection in camelids, because that's more or less what Esther was saying, no, that I will be focused in these 7.2 reservoir host infection models, but with Albert, that if you have seen, he's very interested because he's connected 3 times. So he will, I'm sure, no, this is a joke because I think that also Professor Kim Segales is connected, and you will see later on in the presentation. We have a quite new postdoc that he's mainly doing a lot of organoids. So I will talk a little bit also about the task 7.5 because we are developing organoids. So I guess they will be also willing to answer your questions. So I will start summarizing all the live animal studies we have been done. So as I told you, I'm not gonna enter into detail. So first of all, and as you know, our first studies with MERS coronaviruses were performed in dromedary camels together with other European project, the ZAPI project. But of course, it's not very practice to work with dromedary camels under BSL-3 conditions. And moreover, we also wanted to know if other species were susceptible or not. So we did a livestock susceptibility analysis study to know if other species are susceptible and also if we can use it as a surrogates. So yeah, alpacas and llamas can be used as surrogates, and we use them to study the pathogenesis. And then also we developed a llama direct contact transmission model. So besides the direct inoculation, we also wanted to know this transmission model to mimic more the natural conditions. And mainly these models, both the direct inoculated animals, but also of course the transmission model, allow us to study. So of course, transmission, difference between transmission of different clades of MERS coronaviruses circulating strains. Also this efficacy of MERS vaccine, and then the immunopathogenesis, what Esther says that I will be presenting of MERS in the reservoir, in the natural reservoir, camelids. We did it mainly in alpacas, but also as these animals are not only because they are expensive, but also for ethical reasons, we were using some of the animals that we were using for the transmission studies or the vaccines, also to study more about this immunopathogenesis. These two guys, they are now doctors, is Jordi Rodon and Teniger. Their PhD thesis were partly supported by VetBioNet, and all these studies were part from their thesis. So this one I presented in the last VetBioNet meeting in Brussels. So just to conclude, this is one of the examples of the transmission models that we were doing, and mainly what we conclude is that MERS-CoV clade B strain, in this case we're using one from Qatar in 2015, had extended viral shedding compared to an African clade C strain. And this can be one of the reasons why, for example, in the Arabian Peninsula, this clade B strain is more predominant, so because they outcompete. And also what we conclude with this study is that it's very important to avoid the importation of animals from the Arabian, for example, to Africa, where no human cases have been reported until today, and maybe this can be also one of the reasons. This study, we are also very, very proud of develop some tools for this type of animals, because of course this is the second part, to study the immunopathogenesis of MERS in camelid species. This work I presented in Madrid, so a few years ago, and we developed some tools to assess, to quantify, measure cytokines in camelids. So it was a fluid assay. And we are also very happy that finally we were able to write the manuscript. So it's already in bioRxiv. In fact, we did everything and finished in 2019, but with the pandemic, we were very focused with SARS coronavirus 2. So the manuscript was kind of stopped. And now finally we were able to write it. So just to summarize, and this is a very nice work that Teniger did during his thesis to know more about the immunopathogenesis of MERS. So we knew that experimental infected camelids, they clear the virus within 1 week after developing an immune response. So we wanted to monitor this response with the tools that we developed. And just very briefly, because we studied different parts of the respiratory tract, not also the nasal mucosa, but also trachea and lung. But what we saw is that 2 days post-infection, which is the peak of infection in camelids, in this case in alpacas, type 1 and type 3 interferons were transcribed only in the nasal mucosa, not in the other parts of the respiratory tract. And also interferon stimulated genes, which were also induced in other parts of the respiratory tract. Also in the nasal mucosa, there was an upregulation of the anti-inflammatory cytokine IL-10 and dominant transcription of some of pro-inflammatory genes. So what we conclude is that nasal mucosa is the main target. Well, this we knew, no? It's the main target of MERS coronavirus in camelids, the upper respiratory tract. And then that they are, so why? No, probably one of the reasons that the animals are not suffering from the disease is because they have this efficient innate immune response, no, mainly based in interferon stimulated genes, and also in this dual anti-inflammatory effects of type 3 and type 3 interferons and interleukin 10. And this I presented also in Brussels. So as I told you, we tried to take some samples from experiments. So what we did is to study lamb alveolar macrophages and also lymph nodes to study if they can contribute to the clearance of the virus. And then what we saw, also with the tools that we developed, is that alveolar macrophages can capture, internalize, and degrade viral particles, but they do not induce pro-inflammatory cytokines responses upon viral sensing. So importantly, after despite interferons were induced and also in the lymph nodes, there was an absence of inflammatory responses. And then the last part, so we also tested vaccines, and I'm not gonna explain in detail these vaccines. This is also kind of collaboration with the other European project I was mentioning, ZAPI project, but we were testing different vaccines in this case. This is a multimeric protein scaffold particles based on the RBD part of the vaccine. And in this case, for example, we show that and this was using the transmission model that I was mentioning before. So this vaccine had limit capabilities to avoid the transmission between llamas because it only prevented the infection in one of the 3 vaccinated animals. But then we were testing recombinant S1 vaccine in the same animal model, so in this transmission model. And in this case, we saw that in red are the vaccinated direct in contact animals. So only one of them, we detected viral RNA at different time points after infection, but we could not detect infectious virus in any of the vaccinated animals. And moreover, they had a very good, all the vaccinated animals, they had a very good antibody responses and also checking the neutralization capabilities. So in this case, this vaccine was able to block MERS transmission, which is the objective when we are thinking on developing vaccines against MERS coronavirus. And then, I will try to be very brief. As I told you, we are also very focused in this alternative in vitro ex vivo models. And in fact, I also presented this study. It was in Madrid. We were developing or doing explants from, in this case, alpaca, tracheal explants to test the infection, if it can mimic the natural infection. So of course you are using animals, you need animal to have these explants. But for example, in the case of trachea, llamas has a long neck, so you can have plenty of explants and test many compounds or whatever, with one single animal or 2 or 3, but you don't need plenty of them. But we are also very interested in this part, no, in organoids. Here you can see Dr. Ferran Terrés Freixas. He's the guy I was talking about, our new incorporation, our new postdoc. And he's mainly dealing with developing organoids from different species. Here, I asked him to give me some pictures, some nice pictures about the organoids. Here you can see on the left, chicken lung organoids. On the right side, swine lung organoids. But we are interested in developing from several species. Also, no, there are, because sometimes it's difficult to infect directly organoids. So we are also, for example, in this case, you can do 2D organoid cultures. This is also interesting to see if they keep the structure or not. So first of all, when we start to develop organoids, the first thing we need to do is to validate these organoids if functionally, if structurally, sorry, they are okay or not. So that's what we do. And then we want to validate with infections as well. For example, no, in this case, this is an in vitro infection with avian influenza virus, but we will see other pictures. Here, for example, no, what we see is the structure of these organoids with the tubulin. You can mark the cytoskeleton, or also know which type of cells are there, if are basal cells or which type of cells you have, to really see which structure you have from your organoids and if it's valid or not to do what you want to do. Yeah, here Ferran did an experiment with an avian influenza virus in both organoids, the swine and lungs. So and of course also doing multiple staining, to see the structure of these organoids and if they were infected or not. So this is all work ongoing, and we have worked for a lot of years, I think. It's also interesting, and this is more focused in depending also in the virus or the pathogen you want to test. In this case, influenza. We wanted to know if these organoids had the receptors or not. So alpha-2,3, alpha-2,6 sialic acids, and then check it as well. So mainly, well, we are happy because we can develop organoids, and we have these chicken large organoids. We can detect different population that at least has basal and ciliated cells, so which is good. Both organoids are susceptible to infection, to avian influenza infection, in this case, an H1N1 strain. And then, yeah, as I told you, we did the validations of the different type of cells with these antibodies, and this is a work that is still ongoing. And also, for example, which probably is expected, that swine bronchial organoids express more 2,6 sialic acids than the chicken lungs. So, and in fact, it's true that VetBioNet is finishing. We are discussing how to continue, but we get a project that is specifically to develop a biobank, an organoid biobank from multiple species. We started with swine and chicken, but already Ferran is working on other species, even wildlife species. When we have the chance, we are collaborating with wildlife centers and with the zoos when they have an animal that they need to kill for ethical reasons or for whatever. So we are trying to generate this kind of biobank that we hope and think that will be very useful. And that's it. Many thanks to everybody, especially to, of course, to VetBioNet and to Dr. Albert Ben Said, that we all miss him during this meeting. And yeah, many thanks to all of you.
Thank you, Julia. If there is one question, just one question to go to have a lunch.
Well, it's just more of a comment. So for your vaccine candidate, have you engaged any regulatory advice?
Sorry, what?
Have you engaged any regulatory advice for your vaccine development?
Regulatory advice?
Yeah.
Yeah, if I'm not wrong, this vaccine had, at the moment we test this vaccine, I'm not going to say there was no interest on MERS coronavirus, but it was really difficult not to go to these regulatory things. Because of the COVID, it gained some interest, and now it's in phase, not sure if the last one, but yeah, we are not in, let's say, but CEPI was very interested and found it also this.
It's a perfect time to engage Isidore for free regulatory advice, and we can provide that for you. So you have shameful promotion, I know, but it's important. And the second, your model could be a nice topic for a JRA. I don't know if it's already been put in there, but yeah, lovely stuff.
This will be nice because someone was asking me if we are still interested in MERS coronavirus. We are, but it's true that currently we don't have a project or money to continue. We have to do SARS coronavirus 2 studies, but not in this field. So this will be very interesting. So we'll, yeah.
Well, thank you, Julia. Thank you, everyone. Susanne, I think that you are connected. We have to leave your presentation for after lunch. Is okay for you?
Yeah, so what time would that be then?
What time? 1:30 or?
10 to 2. Yeah. 10 minutes to 2. It would be at 10 minutes to 2. Would that be okay for you?
Yeah, sure. Yeah.
Yeah, okay. Thank you so much for your flexibility and sorry for that.
It's okay.
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 See you.
See ya.
Well, and this the same goes to all the other online participants. Okay, we will be back 10 minutes to 2. Thank you for your understanding.
Ponla, pero ahora, o sea, no importa, no interfiere con la presentación de Susana, que es la que le toca a Susana. Susana primero, que la podríamos pasar por aquí.
One question, Max, if it's possible, just one question.
No, okay.
Está abierto, sí.
Sí. Sí. We start the next part of the Work Package 7 session. We start with the presentation by Susana. Hello, Susana.
Hi.
Hi.
Shall I start sharing?
Are you ready to start
Yeah, sure.
the presentation?
Yeah, I'll share.
Thanks again that you stayed with us. Thanks again that you stayed with us.
No problem.
Yes, thank you very much.
Okay. So do you all see the screen without the... Oh, I can put it smaller, I guess. Yeah, okay, great. So I'd like to continue with our work package 7.3. And it's on the airborne infectious disease models with the topic improve current knowledge of respiratory infectious disease development and spread. And our focus is on infection and co-infection in host with a variable perinatal background. So that means that there's a different exposure during birth and the first days after birth. I want to focus on 2 pathogens. The first one is the PRSV, the porcine reproductive and respiratory syndrome virus, which is causes a high economic burden in the field. And Streptococcus suis, a bacterium that is carried by a lot of piglets, and it colonizes the tonsils. And it does not immediately lead to disease, but different stressors can actually activate the disease. And we want to study these pathogens in 2 different models. The first model is the conventional pig with a known SPF environment. And here I use my pointer. The piglets are born by the vaginal route, so they have perinatal microbial colonization, and they obtain colostrum, so there's the presence of maternal immunity. On the other side, we have a different model. So these are the CD piglets, so cesarean derived and colostrum deprived piglets. And here you see that they are born by the C-section and they are deprived of colostrum. So they have no exposure to the pig microbiome, and they have no maternal immunity obtained, and they are known to be highly susceptible to infection. And this is of interest for vaccine studies. So our aim is to gain knowledge of the model host susceptibility to infection. So for this, we had our study design. We have 6 different groups. First, we have a separation of the CD/CD groups in PRSV and S. suis co-infection. CD/CD animals that only received Streptococcus suis, and of course CD/CD animals that were used as a control. And we had the same for the conventional groups. And this is our timeline. So at day 0, the PRSV groups received PRSV infection. And after 13 days, we collected blood and RNA from the blood. And on day 14, the groups that should receive Streptococcus suis received Streptococcus suis. And on the same time point, we also collected a number of piglets from the study for necropsy. So we had the control animals and the PRSV infected animals. 2 days after Streptococcus suis infection, we again collected blood and RNA. And at day 20 was the end of the study, and the animals were used for a necropsy. So first, we wondered if there is a difference in response to the Streptococcus suis only infection. And we used a bit lower of infection dose that would normally be used because we want to have a nice window to see a response, and we expected the CD/CD piglets to be more susceptible. And indeed, we indeed see that there is loss of survival within this group. So in the black, you see that the conventional animals survived up to the end of the study, and the CD/CD animals reached HAP at day 17. We also tested for spread of the Streptococcus suis. So we swapped different body sites after they got out of the study. So we looked at different sites in the thoracic cavity, the abdominal cavity, different joints, and also the brain. And of course, for the negative controls, we don't see any suis positive cultures. For conventional animals infected with Streptococcus suis, we only see 1 that was positive, but the rest seemed to contain the infection, whereas the CD/CD animals show a high spread of the Streptococcus suis. And this is in line with other measurements we did. Here you see the temperature over time, and upon a suis challenge, you see that the CD/CD animals respond and show a very high increase of the temperature, whereas the conventional animals seem to control this infection. And again, when we also looked at clinical signs, we saw that the CD/CD animals developed clinical signs already at day 15, so 1 day after infection, and they reached the human endpoint at the day 17. But there were no clinical signs at all detected for conventional Streptococcus suis infection animals. So we see that the conventional pigs are able to control the Streptococcus suis infection, and the CD/CD animals don't. So we say here that the birth route or the colostrum intake actually impacts the Streptococcus suis vulnerability in the pigs. So what about the PRSV infection? So for PRSV, we didn't expect to see drastic loss in survival. So we measured here for temperature. We looked at the conventional animals first, and we don't see any response over time that is induced by PRSV. For CD/CD animals, we only see a slight increase which fades over time to baseline. And this is in line with the respiratory signs. So we didn't find any respiratory signs that relate to PRSV infection in both groups. So up to day 13, we didn't see any respiratory signs in the CD background or in the conventional background. And in addition, we couldn't find any difference between the viral load of the PRSV in serum of the animals. So we also measured that over time, and they are equally infected and contained. In addition, we also looked at the lung weight and histology of these animals. And here you see an image which is representative of the lungs that we see upon infection. So here's the lung weight to body weight percentage, and you see that for the control groups, so for CD and the conventional, they're quite equal the same weight. And when you expose to PRSV, you see that the lung weight raises a little bit, but there's not a large difference between the populations. And when you look at the histology images, we see no differences between the CD and the PRSV. Here you see an image that shows consolidated lungs. So you would expect to see open areas like a bit over here, but they're all completely filled, and you see an influx of immune cells in the presence of PRSV. And here you see an increase of macrophages and lymphocytes in the tissue. And still at the day 14 after infection, we see positive cells with PRSV. However, one specific phenotype occurred only in the CD background, and that is the development of red ears, which is specific for the PRSV infection. So here you see one piglet that has the red ears. We didn't see any development of the red ears in the conventional background, but only in the CD background. So after this, we looked into detail in the cell population of the lungs. So we performed a broncholavage of the animals, of the lungs of the animals 14 days after the infection. And first of all, of course, we looked at macrophage populations because the CD163 is the target for the PRSV. So you see in the animals that were not infected, so in the CD animals over here and the conventional animals over here, you see a high presence of macrophages within the CD172 positive population. But upon PRSV infection, you see a drastic drop of this population, and it occurs in the CD background as in the conventional background. On the contrary, we actually see an increase of neutrophils upon a PRSV infection for both the CD animals and the conventional animals. So these were the antigen presenting cells, but now also look at the T cells. Here you see the proportion of the CD3 positive cells that are CD4 cells. And you see that there's hardly any difference between the two groups with or without PRSV. But strikingly, when we look at the CD8 T cells, we see that the CD8 T cell population in the CD background, so without infection, is significantly lower compared to the conventional animals. However, upon a PRSV infection, you see that both the CD animals as the conventional animals are able to increase and respond to this infection to similar levels. So we see that CD animals in the presence of PRSV develop red ears, which is not observed in the conventional background. We also see that the CD animals have less CD8 T cells, but this can get back to conventional levels after PRSV challenge. So what does this PRSV infection then do on the development of Streptococcus suis? Here we see the conventional animals. Again, if you look at the Streptococcus suis, we didn't see any clinical symptoms or loss of survival. But when you first expose the animals to PRSV, we see that Streptococcus suis infection is worse, and we see a loss. In survival. And although the CDCD animals are already more susceptible to Streptococcus suis, we see that the PRSV infection actually aggravates this disease even further, which is still significant. And we see this back when we look at the spread of the Streptococcus suis. So we also analyzed the blood 2 days after Streptococcus infection, so at day 16. And we see that in the presence of PRSV, only a few animals start to show spread to the blood. And although the CDCD animals were more susceptible, this even increased if PRSV is present. And also, again, here I plotted other sites that were sampled as well. And it is most strikingly to see that in the presence of PRSV, we see systemic spread of the Streptococcus suis in the conventional animals. So we see that the PRSV infection promotes systemic spread of Streptococcus suis. So now we've seen that the Streptococcus suis shows quite some difference between CDCD animals and the conventional animals, but PRSV is less clear. So we wondered whether we could see a differential host gene expression in the blood profile. For this, we analyzed the expression of 80 genes that are related to different immune regulatory clusters. So we looked at genes that are associated to antigen-presenting cells, acute phase inflammatory response, anti-inflammatory response, T-cell immunity, TLR expression activation, and endothelial markers. And we analyzed specifically the RNA expression profile that were obtained at day 13. So we analyzed 4 different groups. So we had CDCD and the conventional control groups, and CDCD PRSV infected, and a conventional PRSV infected animals. So first, if we analyze the control groups, we already see that there's a big difference in expression levels of a number of these genes. So here you see the log2 fold change, and I set the cutoff at 1, so there's a doubling at least of the gene expression. And here you see the false discovery rate. And the conventional animals show a high increase of expression of interferon gamma, granzyme A, and the CD1A cluster of genes. But what if we look at the PRSV infection? So these are the results we get when we compare the conventional animals with or without PRSV. And we actually see only 3 genes that pop up significantly induced upon PRSV infection. And I have to say these are after 2 weeks of infection. So the first onset was in the first days. So this is at a later time point during infection. So this is actually what you would expect to see. However, when you look at the PRSV infection in the CDCD background, we see that there's a way stronger response and showing in different differential expressed genes compared to the conventionals. And the 3 that we found for the conventional PRSV infection, we also see back in the CDCD background. So that would mean that these are background independent in response to PRSV, but still we see a specific response to PRSV in only the CDCD background. So for now, I summarize these data, and we see that the cesarean birth and or the colostrum deprivation reduces the control of Streptococcus suis infection. We see red ear phenotype only in the CDCD PRSV background. The CDCD piglets show a reduced CD8 T cell population prior infection, but this can come back to conventional levels after a PRSV infection. We also see that the PRSV infection aggravates the Streptococcus suis infection, which is independent of the perinatal background. So it occurs both in the CDCD background and the conventional background. And we see a CDCD specific enhanced RNA expression profile in response to PRSV. So now we're looking into more detail. And of course, one of the most important things is the microbiome composition, because that's a big difference in these populations. And Paul Stegen will tell about that a bit later today. And we also look into more detail at the local sites of infection, and we're performing RNA sequencing over there to see if there's any barrier disruption. We do histology now and also some serum cytokine profiling. So I would like to thank my colleagues for this work, specifically Sandra, Manouk, and Norbert, because they were a big part of this study. Yeah, that was it.
Thank you very much, Susanna. Quite
You're welcome.
interesting talk. We have time just for one question. Any question? No? Well,
what do you think is the mechanistic background for the red ear phenotype?
Yeah, so what I think, so this red ear would be assuming that the vessels are more dilated, sorry. So I think there is a difference in the endothelial layer or regulation of this. So that it is an important observation that is a clear difference between the populations, whereas the rest remain less clear. Yeah.
I think if I saw it right, you had VCAM upregulation.
Exactly. Yeah.
Also.
Yeah.
So it might be endothelial.
Yeah. So we saw
activation that you see there. Yeah.
Yeah. So we saw VCAM and also occludin. So I think there is a key regulation that is different between those animals groups. Yes. Yeah, for sure.
Okay. Thank you.
You're welcome.
thank you, Susanna. Again, we pass to the next talk that is focused on the fish animal models. This talk will be given by my colleague Patricia Diaz from INEA.
Thank you, Esther. So I think I'm the only fish survivor today. So thank you for staying here. So I divide my presentation in two different parts. The first one, I'm going to explain exactly the methodologies that we have implemented in my group. And at the end, I'm going to show you the last work that we have just published in the frame of this task, in the frame of the VetBioNet. Yeah, so it's only that. And here the point, okay. So I think I'm going to be, I don't know, because here is the camera. Well, I don't know. Anyway, so I titled the talk, Analyzing the Immune Response to Rainbow Trout Pathogens. So I belong to the Animal Health Research Center, as Esther already told us. I belong to the Fish Immunology and Pathology group led by Professor Carolina Tafalla. And our fish model is rainbow trout, Oncorhynchus micis. So we have animal facilities at the level, biological safety levels 2 and 3. So we can work with any kind of fish pathogens at level 3. And as you can see here, so we have, this is the installation, I mean the facility. So we have different tanks with different volume capacities. So we can work with any kind of fish size, depending on the assay that we want to carry out. Our main infection models include virus, like viral hemorrhagic septicemia virus, VHSV, and infections pancreatic necrosis virus, IPNV, and bacteria, Pseudomonas salmonicida, Lactococcus garvieae, and Yersinia rukeri. In this table, I wanted to summarize exactly the different experimental infections that we carry out in our group. So with different fish size, the mode of the infection, the inversion, cohabitation, the sample that we take, and the different sample points. So any researchers that are involved in the VetBioNet can, I mean, they have this, I mean, they can use it. So to study to evaluate the immune response that trigger the viral and the bacterial infection, we have implemented different techniques. So within the molecular techniques, the first thing that we do is real-time PCR, so we can evaluate the different gene expression of different genes involved in the immune response. We are conducting now different microbiome sequences. So we want to evaluate the difference in the intestinal microbiota, depending on the diets, the supplementation of the diets, or even the effect of vaccines. We have carried out a repertoire of immunoglobulins in rainbow trout. For the first time in teleost, we were we analyze the pattern, the transcriptional pattern of a single B cell. Our work is based mainly on flow cytometry. So with that, we are able to characterize the different leukocyte subsets. We are really mainly focused on B cells. So we want to see the capacity of these cells to antigen processing, to proliferate, to phagocytize. And we have as well sorter, so we can really check for example, the transcriptional patterns, the transcript of different separated cells that share the same homology. We can characterize as well the different leukocytes by confocal microscopy. And now we are optimizing protocols to study the binding affinities, the interaction between molecules through the biocore. In the frame of this project, the VetBioNet, we optimize an ELISA to quantify antibodies specific against VHSV and IPMV. We are able now to quantify B cells that secrete antibodies specific to VHSV and IPMV. B cells that are secreting IgD or IL-4, IL-13. We obtain and purify a monoclonal anti-IgT that is suitable for immunohistochemistry. And one of the main goals of our group is to study the differentiation of the B cells to plasma cells, because it's really important to see how the humoral response is going on. So for that, we purify as well an anti-CD38A that we propose, we really confirm that can be a marker for the plasmablast cells. And this is one of the last work that this is another tool. So if the cells are small cells with reduced endoplasmic reticulum, we prove that there are undifferentiated B cells, but if we see always by flow cytometry that the endoplasmic reticulum is expanded and the cells are bigger. So we are they are plasmablasts. So it's another tool to really mark the differentiation. And this is the work that we have just published, is the use of different cell lines to really see the response to viral stimuli. So because you know that the mucosal surface are the main portal entry for virus to the host, and we have available two different cell lines. One is from intestine, the RT-GAT-GC is from rainbow trout, and another as well from the rainbow trout that is from gills, RT-gills. So the idea is to simulate the two different cell lines with the two viruses that we have as a model and with the poly-IC, and analyze the transcriptional pattern and the cytopathic effect. So after the stimulation with the poly-IC, that is the first thing that we did with different concentration of poly-IC. So with after 24 hours, so we analyze different genes that are involved, for example, in the antiviral response, like interferons, or antimicrobial, that is the cathelicidin, or different, the IL-1 beta as a marker for the inflammatory response, or different innate receptors like the TLR. So in general, I'm not going to get bored with that, or because we are on hurry a little bit, so I'm going to be quick. So the main results that we got is that the in RT-GAT, most of the genes that we analyzed were upregulated, but not in RT-GIL. The next thing that we did, we wanted to combine poly-IC with the virus. And again, we got exactly more or less the same results. So in RT-GAT cells, the genes were upregulated stronger than in RT-GIL, and were the same genes that we analyzed that before. Then we want to check to prove really visually that really the cytopathic effect, if the virus can replicate inside the cell lines. So we check by microscopy after 1 day, 2 days, 5 days, and 7 days. Yeah. So we see that both viruses are completely able to replicate in both cell lines. And we see here at 7 days that even the effect of IP and V was was stronger in RT gills. And the last thing that we did was we wanted to really see if the pre-incubation with poly-IC was able to limit the viral replication. So we combine both. So we pre-stimulate with poly-IC, and then after that we we infect it with the virus. And we see here with the RT-GC with the VHSV was a reduction of the viral titer, but was really drastically, I mean, really like more than 6 logs, the viral replication of the IPNV after the poly-IC pre-stimulation. And in gill, the effect was really slightly reduced. So the conclusion of this work was the first one, so the transcriptional response of RT gill cells to the double-strand RNA or to viral encounter was much lower than that of RT gut cells, possible as a consequence of a less effective interference system in RT gills. And the poly-IC almost completely blocked IPNV replication in RT gut cells, but had only minor effects in VHSV replication. So really the important thing is like the use of these cell lines to investigate mucosal immune responses in fish, because they contribute to a further understanding of how they respond to viruses. And thank you for your attention. I try to be quick.
Thank you, Patricia. Time for one question. No? Well, I have just a comment also to highlight the objectives of the Work Package 7. And with this presentation is illustrated the connection between the animal infection models in live animals to the in vitro systems in order to try to comply with the 3 Rs rule. So it's one of the objectives of this work package. Thank you. Next presentation is the last one of this work package, is Tobias May, that is connected online. Hello, Tobias, and thank you for your passion to arrive to this point. Thank you very much. Tobias is going to talk about in vitro models based on immortalization cells.
Thank you, thank you. Do you see my pointer moving around, or?
No, he's not sharing the screen.
I'm not sharing the
screen? We don't see your screen.
Oh?
I can see. No, I see you, but not your your computer screen.
Okay. Okay. So let's see whether this works.
Okay. Now it's
Now,
correct. Thank
again,
you,
the question. So now you see the screen, and do you see also the pointer?
Yes.
Okay, perfect. The question or the scientific question that we were asking, or the thing that we wanted to develop is or were novel in vitro cell systems that are easy to handle, allow infection of relevant pathogens. So in this case, we wanted to create animal cell lines, obviously, and these should be super similar to the in vivo situation, so that you can, in the long run, reduce the need for animal experimentation, or at least, reduce it. Initially, we sat down together within the consortium and discussed which species, which cell models would be of interest, and we came up with different ideas based on the of the importance and also on the availability and the purity of the primary cell systems or the primary cells. So we included different species like sheep, chicken, bovine, and pig, and look for immortalization of endothelial cells, CD14 monocytes, lung epithelial cells, fetal hepatocytes, placental cells, and primary tonsil cells. And this was done, so the primary cells were kindly provided by these different partners here. I hope I did not forget anyone, so please excuse if you're not on the list and we worked with you, sorry. It was a fantastic 5 years, but my memory is not that good anymore. So how do we want to establish novel cell lines? So we have created, obviously for the human system, so for human primary cell, a technology which we call CI-screen. It is based on a gene library. And this gene library is composed of 33 different genes, and these are randomly transduced into the primary cell. And then simply by expanding the cells, we select for those genes that have received an advantageous gene combination, and they proliferate stronger than the untransduced primary cells. So we can then create a lot of cells instead of few cells with a primary cell material. So this is the theory, works nicely for human cells. And in this project, we learned that's also mostly working for, in most settings, working also for animal cells. So the first thing, as I mentioned, is immortalization. So we screen for gene combinations that support the proliferation, and all the other gene combinations we don't see because the cells simply die off. And then the second step is we end up with different immortalized clones. So to simplify it here, I just put 2 clones here and then ship it or then characterize it for gene expression. So to compare it with the primary cell material for protein expression or functional analysis. And with all this cherry picking, you end up then with a optimal cell line meeting your criteria. So in this VetBioNet project, we did the immortalization, and the characterization was done with by the partners, by the collaborators, which provided the primary cells. So one of the questions for us was whether we can also immortalize cells that are not in the mammalian sphere, but like chickens or other birds. So and we tried with chicken different cell types, like embryonal fibroblast, adult fibroblast. Sasha, the lab from Sasha provided us endothelial cells. And with another customer, we tried 2 different cell types. And what can I say? Nothing worked. I know that we can transduce these chicken cells, so it's not a matter of transduction. So I don't know why we can't immortalize these chickens, these primary chicken cells. So it would be interesting to investigate, but there are also a lot of other interesting things, and therefore we haven't looked into it. So it could be, for example, the gene expression cassette, so that the promoters are not super active in chicken could be also that the oncogenes that we used or the expansion genes are not well suited for the chickens. So this is something which we might have a look into at a later stage, but at the moment we don't have capacity for that, unfortunately. What we also did, and this was done in collaboration with Modern, I don't know whether this was correctly pronounced. It was done with Gary Entrican, who was with VetBioNet in the very beginning. He supplied us monocytes from sheep, and up till now or up till then, we didn't know whether we can immortalize cells from the blood system. And I can tell you, yeah, we can now transduce them. That was new, that we did not achieve prior to this project, but we did not succeed in immortalization. So here is this is even more strange, because we get into the cell, obviously into the cells, but we cannot immortalize the cells, because the species cannot be the problem, because when we did the project with Norbert and Paul from Wageningen, we received hepatocytes and placental cells also from sheep. And these were really readily immortalized. So for the hepatocytes, we immortalized 7 cell lines, placental 15 cell lines. And these were not clonal, but polyclonal cells, but they lived happily ever after. As I mentioned, so we did the immortalization, the important and interesting and also time-consuming, laborious stuff was done by Paul, and he in his lab they used this novel model which they called OH3 cell line to reverse engineer these autobunia viruses, and they identified that the viruses require to knock down via the NS gene or protein the interferon pathway, which is shown here. So if you knock it down, you have no replication, and you don't see it in the good old Veros because they don't have an active interferon pathway or signaling. So we can immortalize cells from sheep, but not the CD14 monocytes. We don't know yet why that is. We are now working on the humans monocytes to see whether we can immortalize those. And once we succeed with that, we will go for, I think, porcine monocytes, because we got their collaboration with Manuk and also from Wageningen. But this is still ongoing. Another species, another cell type, and another collaborator, Ignacio or Nacho, I hope this is correct, and Sasha, they provided us porcine or pig lung epithelial cells, nicely differentiated on cell culture inserts. We immortalized them, and they did really nice characterization of these cells. So these cells are able to produce a functional barrier, and when cultivated under under ALI conditions, so air-liquid interface, they differentiate into goblet cells, and also, so shown here with the mucin expression, and also into ciliated cells with the acetylated tubulin expression. They used this model system, plated the cells on cell culture inserts, stimulated them with flagellin, among other stimuli, and then looked for translocation of NF-kappa B. And this can be nicely visualized simply by the nuclear translocation. And this is a transient thing. So you can then simply measure or calculate the cells that show NF-kappa B nucleostaining. And this is what they did here. And after a certain time, in this case 100 minutes, the nuclear staining is again back to 0. And the good thing here is, without going into details here, the good thing is that the phenotype that they observed with the primary epithelial cells and the immortalized cell line is nearly the same in response to flagellin. So it might be that it's slightly more susceptible, but the phenotype is really comparable. So this is really nice data. And another thing that they sent me here, and this is also important to note when you're working with immortalized cell lines, not only cell lines from us, but also with other cell lines. It's important how you culture the cells. And the exact same cell line cultivated under non-differentiated conditions versus differentiated conditions, and infected with influenza virus. And you see that under non-differentiated conditions, you see nice infection, not 100%, but really nice infection, which is not seen in that amount under differentiated conditions. And also, this is just a small snapshot of the work that they did. The cytokine profile. So everything's going up in response to this influenza virus, but the magnitude of induction differs a lot when you compare, for example, CXCL2. It's 30-fold induced in the non-differentiated versus 5-fold in the differentiated. Others are similar. So you really have to look and you have to know your model you're working with and then draw the correct conclusions. So really nice data. Another approach, this was done in collaboration with Ferdinand Rösch. There we wanted to create in vitro model for AFSV infection, so African swine fever, isolated palatine tonsils, received a mixture of primary cells and immortalized this. So didn't care about the primary cells, the isolation of the primary cells, but did a immortalization, a crude immortalization of the different cell types. And just by looking at those different cell lines, you see that they show a complete different morphology and thus will be of different cell type origin. And the reasoning behind it was to identify or hopefully identify the cell type which supports the replication of AFSV. Unfortunately, so this is the best one, and this is in comparison to the monocytes. So it was so the cells look really nice, but they do not support the proliferation replication of ASFV to an extent that is desirable for an in vitro model of for an in vitro model. So that's coming really in a brief run through the things that we did and we tried. So we can create novel in vitro models all from in the mammalian space if they are adherent. So if you are a chicken or if you are lymphocytes or monocyte, we won't be able to succeed in immortalization. So these cell lines can be useful tools. But yeah, sometimes, like the last story, you have to find the right virus to support the replication and to study the things. And importantly, always know your in vitro model as good as you can, because then you can really get nice results out of it. And we in this project we had really the easiest part, because we only had to immortalize the stuff. The real work started when we handed over the cells to our collaborators, and they did a fantastic job, and I want to thank them for this. And I want to thank a big thank you to all of you, because it was really a pleasure working with you. Unfortunately, I couldn't come and see you, but I'm pretty sure we will meet and then have a nice glass of wine, beer, whatsoever. So if there are any questions and if there's time, I'd be happy to answer your questions.
Thank you, Doya. Time for one question.
Thank you, Tobias. It's Keith Ballingle here from the Morden Research Institute. I was involved right at the early part of the study with Gary and Sean, and it's obviously a great frustration that that anything that's not adherent struggles to be immortalized. Have you managed to try and figure out why the suspension cells are much more difficult to immortalize compared to the adherent?
Yeah, so there are different layers. So different cell types require different conditions. So for the monocytes, we took the most difficult cell type, because they have obviously, which I didn't know before, they have a protection mechanism in place. So there is an enzyme expressed that allows the transduction of these viruses, but prohibits the integration into the host genome, and thereby they they are degraded, and we cannot stably integrate our gene library. So we are trying to work, and it was shown that if you knock this protein off, out, then you can nicely and efficiently transduce these monocytes. So this is what we are trying now with human monocytes, and if this is working, then we would be happy to move forward. With T cells, B cells, and all these other cells, they are obviously missing a receptor that the VSVG that we use for the lentivirus. So the VSVG targets a certain receptor which is missing on the T cell, B cells, and so on. So if you're using another surface protein as for the lentiviral pseudotypization, then you might end up with a better transduction of these cells. So this is something we will follow. We don't have any data on this. So these are at the moment my explanations why they are hard to immortalize. So I don't think it's a cell cycle, because this is more or less conserved throughout the kingdom. So the animal kingdom, cell type kingdom, whatsoever. So, yeah.
Okay, thank you. Thank you very much.
the last rapid question.
I mean, you already answered to one question. I mean, if you try to pseudotype your lentivirus system. And second, in your library of lentivirus, have you included SV40, large T antigen, or not?
It is.
Okay. Thank you.
Therefore, I was a bit astonished.
Usually works very well. I mean, if you combine large T antigen plus telomerase, works always, but you know.
Yeah, that's why
Yes.
I was astonished that the chicken cells did not make it because I thought, okay, if nothing works, then the T antigen should work, but obviously it didn't help. For reasons I'm not sure, I do not know, because I didn't have the time to really have a look into it. So I know that most of the cell lines I encountered were done with E1A and E1B from adenovirus, which we don't have included in our library. But I don't know whether this is really the cause for that.
Well, so I think that we can, thank you, Tobias.
Thank
you.
We
can conclude the Work Package 7 session. Thank you everyone to be here, and also the people connected, and of course to the speakers of this session. And I say the same that Tobias, that has been a pleasure to collaborate with all of you. And I am going to miss you, because these meetings are very special, and it's a pity. So, well, I hope to see you in a future project again. Thank you very much.
So then I suggest that Ryan will join me here, and then we will open the last session for the meeting, which is then dedicated to Work Package 8.
Thank you. Thanks, Sasha.
Can you wait a second? No, maybe give it to me. Maybe later I am leaving. Okay, okay.
So I can just speak. Try and shave a few minutes off. So I know we're a little bit late, and I've got the last shift. So just this work package 8, is I'm work package leader for that, and it's the title is here, and it's basically to the aim of this. It was a joint research activity, and the aim was to produce and develop novel analytical tools to help with the host virus interaction. There were various topics, various research proposals within this. I've got this here, and so they've been organised this way. Is it best if I stand this side? So we organised this work package, this JRA, into 3 broad topic areas. The first is developing novel transcriptomic tools. Here, and the task leader for the subtask leader for that was Steve Gordon. The second one was to look at develop novel tools to look at explore and dissect the immunological synapse using NGS tools. That was 8.2, and the 8.3 was to use NGS sequencing and pipeline development to examine the viral swarms and microbiome within animals. So this second one basically looking at genomics and bioinformatics, sort of as as the umbrella heading. And the third one was included 2 tasks, both under the umbrella of developing novel serological assays. One was for RHDV, to develop serotyping and differentiation tools to differentiate between the different types. And the second part of this was to look at and to develop as ELISAs for salmonids, so fish ELISAs. So this is it's just an overview of the presentations. I know we're a bit behind, but this is the running order. I'll put myself at the end here, so I'll be able to cut that down if we've not got enough time. But this is the running order of the talks today. And so I guess the best thing is just to get stuck into it. So the first is the first presentations by Ord, and that's discussing the task 8.1, which is looking at these novel transcriptomic tools. So
thank you very much, Ryan. So Steve Gordon was supposed to present, but unfortunately he emailed me last week to say that he couldn't join the meeting. So I'm presenting on his behalf the work that we performed together on this task 8.1 regarding transcriptomic.
This is what?
Yes. Thanks. I will focus on the work that I've performed with Steve on the analysis of bovine lung cells infected by Mycobacterium bovis. So I just wanted to start with a comparison of the technology that were developed in this task. So Steve was in charge of comparing the fluidigm and the nanostring technology. So just quick reminder. So you all know RNA-seq, and we know that costs decreased over time, but it demands a lot for data analysis. The fluidigm, it's easier, but it's more like a medium throughput qPCR. So you have to design primers, and it's time to optimize such tools. And it requires an amplification step, which is nice because you can start with a low amount of materials. On the other side, nanostring, the code set, the probes are designed by nanostring, so you save time and you have minimum optimization. It's non-enzymatic, and it's also work with formalin fixed samples, so that's also good to know. The consumable cost is a bit similar to RNA-seq, but it's the analysis is much simpler. So that's a picture of the instrument. So that's the fluidigm and the plates, and the nanostring, which is at the University College of Dublin in Steve's lab. So that's the subtask that Steve already presented at the Brussels meeting last year. So I'm not going to detail them, just that I was more involved in the fluidigm. So that's what I'm going to highlight today. And I'm going to give you some data on it's kind of nicely linked to our package 7, actually, because we also exploit the precisely cut lung slices model that we are using for cattle lung explant. And what we observed after infection with Mycobacterium bovis. So bovis is the leading agent of bovine tuberculosis. So that disease, we don't know much about the physiopathology. And of course, we lack correlate of protection. And it's the main knowledge gap to better tackle the disease. The early interaction that occurs in the lung between the mycobacteria and the host cells are very critical steps that govern the outcome of the disease. And that's why we are focusing on those steps. And in cattle, those steps are very poorly characterized. So we took advantage of the lung explant model that we developed. And we tried to understand what are those early steps that influence the physiopathology. So we answer several questions, or try to answer several questions. We compared 2 different kinds of mycobacteria. Mycobacterium bovis, which is the causing agent of bovine tuberculosis, but can infect a wide range of hosts, including human, it's zoonotic, and also wildlife. And we also compare MTB, so Mycobacterium tuberculosis, which is the human pathogen, but occasionally can also infect cattle, but it's very rare. So we try to understand the host species preference here in the cattle lung explant. We were also conducted our study on 2 different beef breeds to try to understand if genetic background matters, because we know that for zebu, so both indicus, they are more resistant to BTB than both taurus. So we just here compared 2 different very important beef breeds in France. And we were looking for biomarkers of virulence, and especially the type 1 interferon pathway, because it's very important in human tuberculosis. So in a nutshell, what we've done, and that's more related to VP7 to start with. So collecting bovine lung at different slaughterhouses, we filled that with medium and low melting point agarose, and then use this machine, crumdic tissue slicer, to obtain a lung explant. So there are 8 millimeters round piece of lungs with 100 micron of thickness that you can put in culture plates, and by changing the media and put it back the plate in the incubator, you just remove the low melting point agarose, and you can nicely see the ciliated cells that are moving. So tissue is alive, you correctly remove the agarose, and you can go on with the infection. So then after monitoring the ciliary activity and the viability of the tissue, we entered the BSL-3 lab, and in fact that with different mycobacterial strains. So the first 2 are Bovis strain. So the UK reference strain, a French reference strain, H17RV, it's an MTB strain, a lab strain. And the last one it's an MTB strain, so tuberculosis strain, but that was isolated from a zebu in Ethiopia. So what we've done to analyze them, so of course we monitor cytotoxicity, and I'm not going to present, but viability of the tissue is very good even 48 hours after infection. So I'm going to focus on the work that we've performed with the RNA, so the transcriptomic, but we also done some work on the cytokines, chemokines, and also some imaging. So regarding the transcriptomic analysis, and that's what's fit in this work package 8. So we compared the fluidine technology and the nanostring. So for our system, because of the residue of agarose in the tissue, we first think that fluidine is better, and nanostring needed more or better quality of RNA. But after improving the protocol at the end, we managed to obtain some results with the nanostring with Steve on those lung explants, but it required a lot of improvement of our RNA extraction protocol. So in the lung explants, we know that all mycobacteria reach the alveoli and can be internalized by alveolar macrophages. So that's here. 20% of mycobacteria are inside macrophages. And in alveoli where mycobacteria is present, we have a recruitment of alveolar macrophages 48 hours after infection, proving that this system is very dynamic, and you can also monitor cell movement. Then we analyzed what the tissue was producing, and we compared the 2 breeds, the blonde d'Aquitaine and the Charolaise, and we were really surprised to see such important differences. So that's the principal component analysis of cytokine and 96 gene expression on the 2 breeds. So you can see that there is very distinct profile with the 4 different mycobacterial strain. Regarding gene expression, we have almost no differentially expressed gene after infection with the Charolaise, red triangle here, and a very heterogeneous and various response with the blonde d'Aquitaine. So huge difference between the 2 breeds. So we analyzed a lot of genes related to immune responses and inflammation. And as I said earlier, we focused on the type 1 interferon pathway also with the fluidine. And you can see here on this slide that we had lots of genes related to this pathway that were upregulated after infection with the AF2122 and MB3601, that are the 2 Bovis strain, and almost nothing with H17RV, the MTB strain. And this MTB strain isolated from a cow, we had an intermediate profile. So proving here that this pathway was induced after an infection with Bovis, but not M tuberculosis. And that's the protein level, the CXL10 in ELISA, to confirm that this pathway was indeed induced. Because we saw that alveolar macrophages were uptake the mycobacteria, we were wondering if they contributed to this pathway. So we also performed some experiments using alveolar macrophages from bronchoalveolar lavage, and only with 2 strains, but here the Bovis strain, we see that they contribute to this type 1 interferon signature, but they're not the only one, as we have more genes differentially regulated with the PCLS, the whole lung explant. Again, showing the relevance of this complex model when we have all the same types, connection between cells, and also production of key molecules like mucus, surfactant, etc. So the take-home messages, so with the Fluidigm in the lab, we have designed more than 250 primers to study cattle immune responses. So they are available at INRAE. We also show that the host genetic matters in response to mycobacteria, but now it requires more investigation to see if we can point out why, which gene, and if it's relevant for genomic selection. Also differences in the host response to distinct mycobacteria, and especially this type 1 interferon pathway that was only induced after infection with bovis. And we know that this pathway is very important for the virulence of the mycobacteria in vivo. So we really believe that this lung explant model could help us deciphering biomarkers of resistance and susceptibility to BTB. And we're really glad that we have designed so many useful transcriptomic tools thanks to VetBioNet joint research activity. So that's the people that I want to acknowledge for the results. So people from my lab at INRAE, of course, Steve, who is not here today, but big thanks to him, and also other collaborators from ANSES and other lab at INRAE. And because the purpose today is to show that we work great together, thanks to the VetBioNet connection and also money, we were able to secure other funding. So we obtained 2 bilateral projects to exchange students. It's a PhD Ulysses. So the first one in 2018. So I was able to visit Steve several times, and one of his postdoc, Boyan, came to INRAE. And then we were lucky to have another one in 2021. And this time, 2 PhD students came to INRAE, and my former PhD student visited Steve to learn more about transcriptomic analysis tools. So the work that we performed together was partly published, so in those 2 papers. And I want also to highlight Susana Flores, who submitted her thesis and is now a doctor. She's back to Mexico. That's Susana and I, who presented our results at the International Bovis Conference in Galway last year. And Susana has 2 other manuscripts coming, and they are submitted, so more to come from us and from the Dublin group. And we talked a bit about it yesterday, so we cross our finger for the joint doctoral school JEDI. So thanks to the work that we perform with VetBioNet, Stephen, Gordon, and I we proposed 2 doctoral proposals. So we cross our finger for JEDI. May the force be with us, and by the force I mean may European money be with us for the future. Thank you very much.
Thank you very much, Jord. That's really, really interesting. I was wondering, we've probably got time for 1 question, or maybe 2.
I don't want to be too invasive. So usually when you treat cells infected by mycobacteria with interferon gamma, you kill mycobacteria. Can you do the same with type 1 interferon?
Yeah, we haven't done such an experiment yet, but so regarding type 1 interferon, what is known is it's in the advantage of the mycobacteria because it's reduced the inflammasomes and the production of IL-1 beta and stuff like that. So there are nice paper using mouse model and bovis showing that when the type 1 interferon pathway is induced in vivo, you have more bacterial burden, more pathology, and so maybe if we treat the PCLS with type 1 interferon, maybe we see more CFUs or stuff that we haven't tried yet. It's a very short time, only 2 days for now. We try to have longer experiment now, But remember that the time for division of microbacteria, it's 1 day. So we need to have longer culture, but it's what we are doing now.
Oops, sorry. There's a spark between us. Some energy flow. I have a quick question also. I was wondering if there's any interest to develop the PCLS model also for BTB infections with carnivores, like in collaboration with Sandrine or someone else, with red foxes or ferrets.
Yeah, we are starting to collaborate with Sandrine. We will start developing material for ferrets because it's a nice model for BTB in wildlife. So of course, more to come related to other species than cattle. And it's very easy to perform PCLS on every different species. I've done it in cattle, in mouse, and I'm doing it with human tissue. So I don't see any technical issue. And of course, it's going to be feasible in the ferrets. We just need tools, immunological tools to analyze what is going on after infection. So transcriptomic tools, antibodies, but we are starting to work on that. We just actually are missing very much the ferrets, which, because they're reserved for COVID-19 studies. So it's difficult to have some ferrets for now to work on TB.
I believe there are fluidime assays for minks. I don't know how, but for ferrets, I'm not aware. But maybe during the past years that should have been developed.
Yeah.
So.
Yeah, there are a lot of studies using single cells RNA with the ferrets. So transcriptomic tools are going to develop quicker than antibodies, for instance, and yeah, ELISA and stuff.
Okay, thank you.
Welcome.
Thanks. Okay, I should have just said at the beginning that the talks in this session are examples of some of the work. It's not all of the work that's been undertaken in this work package. Okay, so the next presentations by Ghislaine and Esther, and this is related to the development of antibody-based detection and differentiation of the two different RHDV types. So
Giselle, can you tell me which one is your? Yeah, but can you tell me just the name? It has to be here. No, it's.
Oh, 8.4. It's up. It's there. This one. That's it. This one.
Yes, yes. Sorry, many presentations.
No, exactly. No. It must be hard to keep track of all of them, right?
C'est ça. So I will present this task with Esther, and on the development of new diagnostic immunoassays for RHDV, rabbit hemorrhagic disease virus. And I will begin by presenting the virus and the disease and a few the objectives. And then Esther will continue on the results. So this virus is a calicivirus. It's first detected in 1984 in China, and it infects domestic and wild European rabbits over weeks old. And this virus gives very high mortality levels and a very quick time. And it is endemic in European countries and also in Australia and New Zealand, where it was introduced to control invasive European rabbits, but it's present in large part of the world. And he has a very high economic impact for bred rabbits. And this is why this disease is listed by OIE, now World Organization for Animal Health. Unfortunately, this disease is controlled by vaccination and biosafety measure. Sorry. But in 2010, a new genotype named RHDV2 emerged in France. And this genotype escapes to the immunity induced by RHDV vaccines. It infects European rabbits, of course, but as young as 11 days. And it also infects several continental rabbit species and air species. And this new genotype quickly spread to several countries in Europe, of course, but also in Australia, Africa, North America, and very recently Asia. But since 2016, this new genotype has now mortality levels comparable to that of RHDV1. So it's a very infectious disease. It has replaced most of the RHDV in several European countries, and new specific vaccines have been developed to control this disease. But RHDV immunoassays are not sufficient to detect this new genotype. So it was necessary to develop new immunological assays in order to distinguish the two genotypes. And so at this step, I let Esther to continue to present the results she obtained. No, sorry. There is objectives. So just very quickly, this task involved 3 institutes, 2 Spanish in here and in Genesa. In Genesa, it's a private society, and in France, ANSES. So the first step was to generate virus-like particles from different genotypes, on the 2 pathogenic genotypes, RHDV and RHDV2, and 1 non-pathogenic, the rabbit calicivirus. The first objective was to produce sera, polyclonal sera, by inoculating SPF rabbits with these VLPs, and this objective was realized in the ANSES BSL2 facilities. The third one was to analyze this experimental sera, and a new ELISA test have been developed for that, and to study antigenic variability among the different strains. Next objective was to identify relevant epitopes of RHDV2 and to generate new monoclonal antibodies to develop new assays. And in particular, to detect the new genotype RHDV2. And the last one was to develop an antigen detection test based on lateral flow assay that allow to detect and to differentiate the 2 genotypes. And this assay has been validated with field samples. So thank you.
Thank you, Ghislaine. Yes. So as Ghislaine has introduced the disease, I am going to show you the main results that we have obtained in the framework of this project. Due to the emergence of this new variant of rabbit hemorrhagic disease, type 2, the diagnostic immunoassays, but also those for the direct detection of the virus, were not adapted for or optimized for the detection of these new viruses. So here I am going to present the development of a rapid assay for differential detection of rabbit hemorrhagic disease virus genotypes 1 and 2. As Ghislaine has mentioned, this work has been performed in cooperation with the 3 partners of VetBioNet, INEA, Eurofins Ingenasa, and also ANSES. So the objectives were firstly the development and selection of monoclonal antibodies for a specific recognition of both rabbit hemorrhagic disease genotypes, the development of the duplex lateral flow assay to differentiate and detect at the same time both genotypes, and after to evaluate the prototype developed firstly by analytical sensitivity and also by the diagnostic performance, testing the sensitivity and specificity, testing rabbit samples. The first step was the development and selection of monoclonals. For this task, we produce a recombinant rabbit hemorrhagic disease virus VLPs, virus-like particles. So in our lab at INEA, we have developed a very efficient in vitro system for the production of these recombinant proteins, these particles. We produce baculovirus recombinant that express the protein BP60 that form the capsid of the rabbit hemorrhagic disease, and we can produce high quantities of these particles in insect cells. So as you can see in the Kumasi polyacrylamide gel, the expression of these specific proteins is highly, and after a simple process to purify these proteins, we can see by microscopy, electronic microscopy, the correct formation of these VLPs from genotype 1 and genotype 2. So the next step was to analyze the antigenic differences between these genotypes. So making a PEP scan, we identify a region that was antigenically different between these 2 genotypes. So we defined the strategy for the immunization of mice, firstly immunizing with the VLPs, rabbit hemorrhagic disease virus type 2, and with a boost that using a synthetic peptide that reproduce this antigenic epitope or region that is different between both genotypes. With conventional system, we produce a collection of monoclonals that were screening, cloned, and finally characterized by Western blot technologies and also by ELISA. We obtained 10 monoclonals. 5 of them were specific for genotype 2, just recognize this genotype, and other 5 monoclonals that recognize both genotypes, genotype 1 and genotype 2. After the characterization by in the ELISA and after check the isotype, we select a monoclonal specific for the genotype 2, and we perform immunofluorescence analysis to check this specificity, and this is an example where you can see that this monoclonal only recognize insect cells infected with the baculovirus that express the type 2, but not the type 1. So with these reagents, we selected the monoclonals to be part of this new assay, the lateral flow assay. And also we established the conditions of this duplex lateral flow test as a buffer running and the device. And we start to set up the test procedure. So the first step is the sample preparation. Since this assay is a rapid assay, we thought to use liver, that is the main target for the rabbit hemorrhagic disease infections, testing samples that were homogenized very simple with a little piece of liver in just a buffer of PBS that could be easily prepared, or testing directly exudates that are more easy because it's just to take these exudates from the liver and to use as a sample. We add 20 microliters of this sample, but it's not necessary to the quantification. I think that after the SARS-CoV-2 pandemic, everyone knows how to use this type of test. So it's really easy, no? So, well, 20 microliters should be added in this window for sampling after 3 drops of running buffer. And waiting 10 minutes to read the results, we can find this type of potential results. If the sample that is analyzed is negative, we just see a blue band corresponding to the control. If the sample is positive for rabbit hemorrhagic disease type 1, we see this red band corresponding to T1. And if the sample is positive to genotype 2, I can see the red band in the position according to T2. But sometimes when we have a lot of viral particles in these samples, since the other monoclonal that recognize the type 1 is also capable to recognize the type 2, we can see even 2 bands. Still, 1 or 2 are positive for genotype 2. So this is an additional characteristic of this test, that you can differentiate between both genotypes, and you have also a preliminary idea about the concentration of the viral particles that are in your sample. So with this test, we start the evaluation of this prototype, firstly with to measure the analytical sensibility. This was performed using these recombinant VLPs of genotype 1 in blue or genotype 2 in orange. As you can see, the limit of detection of the assay is lower for the genotype 2. Only the limit is 0.02 micrograms milliliter, and for genotype 1 is 0.08 micrograms per milliliter. But this analytical sensibility is adequate for a diagnostic assay. So we analyze the diagnostic performance of this test, testing 96 liver samples and also 40 liver exudates. These samples were classified as rabbit hemorrhagic disease type 1 positive, type 2 positive or negative using an RT-PCR. And we calculated the sensitivity and the specificity of this test using as gold standard the RT-PCR. As you can see in these tables, the sensitivity testing liver samples or liver exudates for genotype 2 is quite similar. It's around more than 95%. And for the rabbit hemorrhagic disease virus type 1, 91.3. This is according with the analytical sensitivity that we detected. In both cases, for liver samples and liver exudates, the specificity was 100%. So the main conclusions of these results is that we have obtained a set of 10 monoclonal antibodies that have been obtained and characterized. 5 of them are common to both genotypes, and other 5 are specific for genotype 2. Indeed, these monoclonals are being used also for a serological assay for detection and differentiate antibodies against these 2 genotypes. And we have developed the first duplex lateral flow assay that allows the specific differentiation of these rabbit hemorrhagic disease virus genotypes at the same time with the same volume of samples. So it's quite rapid, sensible, and specific. Indeed, future prospects include that last year, when we present these results during the World Lagomorph Congress in Montpellier, the coordinator of an European life project, Life Iberconejo, the coordinator is the WWF Spain, asked us to participate in this project as advisors, and we attend a meeting in Portugal. And in this meeting with the stakeholders involved in the control of this disease in the field, we finally decided to use this system and also the serological assays that we have also developed for the monitoring of rabbit population in the Iberia Peninsula. We think that this assay could be very useful for the passive surveillance. I think that Ghislaine has mentioned that this disease is economically important, but also from the point of view of the biodiversity in the field is quite important, especially in the Mediterranean area, because the rabbit is connected with the surveillance of animal species that are in danger. The other part is like as partners, together with ANSES, of an European project from the Primacol, named LACMED, in collaboration with partners from Portugal, Civio, and also from Italy, and Istituto Profiláctico de Brescia. We are going to use this duplex lateral flow test to validate non-invasive samples. We want to evaluate, to assess the potential of this kind of tests for also to the active surveillance, testing samples that can be collected from the animals' life as feces and saliva. So I think that could be quite interesting for the control of this disease. Thank you for your attention. This is a photograph of rabbits that live at INEA Institute. They are very healthy, so the biosecurity measures are okay, but there is a lot of her. So thank you very much.
Thanks very much, Esther and Ghislaine. That was really interesting. I had one question, if I may. The epitopes, you know, that these antibodies bind to, the specific binding in particular. Do you have any idea if they're linear or conformational? The reason I ask is, if they're more linear, perhaps you could maybe use them to develop a serological LFD to opposed to an antigen, or in addition to the antigen detection for serosurveillance?
Yes, this is correct. It's a linear epitope, and indeed we have a format of ELISA, not in lateral flow. We have discussed about the possibility to develop, the first step was this, but the idea is to develop also this serological rapid test using these monoclonals and this sequence that we have identified.
Okay, was there any other questions? Keith.
Thanks both of you for a very nice presentation. I'm just interested in the origin of the hemorrhagic disease 2. I mean, presumably you have the sequence of the genomes of both. So have you any idea how the second or where the second variant came from? Because it appears to be very different.
Yeah, probably Ghislaine, no? Well,
we have no idea, in fact, no.
Okay, so phylogenetic.
We didn't find the original virus, because I don't know if you know, but the RSGV2 is a recombinant virus between for its structural part a new Lagovirus, and for the non-structural part and non-pathogenic Lagovirus that we know, the that circulate in Europe, in France notably. So, but we didn't find the original RHDV2 non-recombinant one. We don't know.
Interesting, interesting.
Just to add that I think that there is two hypotheses. One is that it's an evolution of what these non-pathogenic viruses, and the other is that it's a new one, that there is a lot of caliciviruses circulating in different populations. So indeed, this type 2 is quite interesting also because in addition to being different from the type 1, has a broad host tropism and has been found in hares. I don't know the name in English. This is not rabbits, because type 1 just infect rabbits, but this type 2 infect hares of different species and has been found also in other sympatric small carnivores in the nature. So it's like increasing the host tropism of the virus.
Okay, thank you very much.
Okay, well, thank you very much for that. So the next presentation, I believe, is on online, and it's from Paul Steger, discussing microbiome, some of the work that's been undertaken looking at the microbiome in pigs. So this is online, I guess. So I'll just hand over to you, Paul.
Thank you very much. Is everything working?
Yep, we can hear you.
Great. All right. So my name is Paul, and I work as a bioinformatician at WBVR in Lelystad. And today I will share with you the progress on this project, where we study the respiratory microbiota in relation to PRSV and Streptococcus suis infection. So over the last 50 years, we have changed our view on the microbiota from something that just happens to be there to something that actually contributes to important functions. And these functions for the gut microbiome include nutrient conversion of feed, training the immune system, and finally also protecting against pathogens that might colonize, in this case the gut, and then cause disease. So this is a form of colonization resistance that it's also offering. Now, by comparing healthy animals and diseased animals, we slowly learned which bacteria are associated with a state of health and which one with a state of disease. Now, this is way more developed in humans, where we, for instance, know that certain toxin-producing bacteria are associated with diseases like inflammatory bowel disease, but also we've made associations with lesser well-understood diseases like Parkinson's, Alzheimer's, and multiple sclerosis, which have now also been associated with some bacteria that might play a role there. So going back to the topic of today, the pig respiratory microbiome. Now, this is a study this is a field that is not well studied. So less is known about the respiratory microbiome compared to the gut microbiota, but some of these same principles still apply. For instance, training of the immune system was found to be important, and also some other qualities. So what is known is that right after birth, the respiratory microbiota undergoes a major transition, where it undergoes massive fluctuations in its composition, and then slowly stabilizes around the period of weaning, where it then forms an equilibrium. But in this first part, the part right after birth, it is very susceptible to colonizations by different pathogens. This can then cause disease. So these include Acetinomyces, Glasarella, and Streptococcus suis, from which this last one is the topic of this project. So Streptococcus suis is a gram-positive bacteria, but the interesting thing is that in adult pigs, it's present as an asymptomatic colonizer, so it's not really causing any disease then. But in these newborn pigs, it is acting like a true pathogen, where it can cross the mucal barriers epithelial barriers, and then cause disease like arthritis, endocarditis, sepsis, and meningitis. On top of all that, it's also a zoonosis, which can spread to humans and then cause disease there as well. So you can see why this is an important pathogen, not just for pig health and well-being, but also in general in the industry. So there's already more known about this pathogen, and also that it's not just the infection itself that's causing symptoms, but it's also part of much larger systemic changes. So we know that Streptococcus suis infection has often coincided with changes in the microbiota composition, that is the respiratory microbiota. And in addition to that, it is coincided with PRSV infections, which is this other important pathogen causing this respiratory syndrome virus. So therefore, when studying a Streptococcus suis infection, we should not just focus on Streptococcus suis and kind of imagine that these other that the microbiota and PRSV are also there, but also take them into account, which is what we have done in this study design, which was already introduced by Susanna, where we in one study design study PRSV infection, Streptococcus suis infection, but also the respiratory microbiota. So then if we go over this design very quickly, we start with 6-week-old pigs that are infected with PRSV. Then 2 weeks later with Streptococcus suis, and then this day 21, there is a sample collection site of both the upper respiratory tract, so nasal and tonsil samples, but also the lower respiratory tract of bronchial and lung samples. So in this way, we end up with 3 groups. We have a control group not exposed to any pathogen, then the Streptococcus suis only group, and the co-infection group, which allows us to determine in which group the microbiota is responding to these specific elements. Now, there's one important factor that we cannot ignore, which is the starting point right here. So these 6-week-old pigs, because we already know that the mode of birth is very important for this starting point of the microbiota, where often in the natural way of birth, the respiratory microbiota matches the vaginal microbiota and skin microbiota of the mothers. So therefore, we go to the complete study setup, where we side-by-side compare the effect of conventional birth to cesarean section and colostrum-derived birth. So these subgroups still remain the same, and this is then our setup. Now, to determine which bacteria are there, we have to sequence the bacteria, of course, and there are several ways to do so. And for this project, we chose to sequence the full genomes of bacteria, which will allow us to determine exactly which species are present. Now, this is different to most of the past studies that have been performed to these kind of directions, where only a small part of the genome has been sequenced, so the 16S rRNA gene, which often does not contain sufficient information to determine which species are there, but can only go to genus level. So this is one of the novelties of this study. And in order to get this nice information, we apply shotgun metagenomics. So this will allow us to receive all of the sequencing data necessary to make these conclusions. Now, to go into the current status of the project, the sample collection was all successful, and you already saw these initial analyses that were performed. But for some reason, the sequencing data was massively delayed by the sequencing companies, and I only received that yesterday. It's 2.5 terabytes of data, so it's really a nice amount. But I didn't get around to look into in the composition of the microbiota, so that's quite unfortunate. But instead, I can use two slides to kind of show our expectations of what we might find. Because one of our colleagues in Wageningen, he performed this as a similar kind of study, but then on one of these sub-questions, where they studied Streptococcus suis infection and in which regard this alters the tonsil microbiome. So this is published only on bioRxiv on the end of last year. And they showed that indeed Streptococcus suis infection is able to alter the microbiome. So you have this setup with a control farm and healthy pigs, and then a case farm with some pigs showing disease and control pigs. And the first thing that they determined is how the microbiota is divided in terms of species richness. So this is alpha diversity. I will come back to this later point later. To alpha diversity, shown over here. And on the control farm, they determined species richness between an index of 4 and 4.5, which resembles a very diverse microbiota. And this is usually related to kind of state of health. This matches the healthy pigs on this case farm. So these have a similar kind of index. But what you can see immediately with these diseased pigs is they have strongly reduced alpha diversity, indicating lower species richness, which is often a sign of reduced health. And the other thing that they included in this setup is that they also studied the relation to siblings of these diseased pigs. And as you might expect, they still shown a sign of an altered microbiota. So apparently these changes are that thorough that they will have an effect on offspring as well, kind of starting with this setback in their microbiota. Now, from this point, from studying which species were present, they switched to changes in the abundance of species. So this is the other result that I wanted to show you. You can see these disease pigs plotted next to the control group, and each dot here represents an individual pig. And on the y-axis, you can see the relative abundance of this Prevotella species. So in these diseased pigs, there's a higher abundance of these species compared to the control group, and this is a significant difference. Now, unfortunately, because of their sequencing method, they're not able to determine which species this is, and because they have used this other method where you have limited information. However, they also found more differences, and with this second one, they have sufficient information to actually determine the species, and it's Fusobacterium gastrosuis, which is present in higher abundance in diseased pigs compared to control pigs. So this is quite interesting because this is a known pathogen, although it's not often able to induce disease on its own, but it kind of requires another pathogen to create an entry in tissue. So this is quite interesting that they already showed these associations, also with this last one, this undetermined Prevotella species. And we expect with our setup to find similar associations, but then with the species level of resolution. So to then come to the summary kind of already, the sequencing results are pending. I expect to work on this coming week, which is unfortunately too late, but I hope I will somehow still be able to show the results. And and secondly, we expect to find similar associations, might even be the same species, but then on species level of resolution. So with this controlled setup and these advanced sequencing technique, we expect to have a higher level of resolution and determine these associations in more detail. So with that, I would like to thank all the people that worked on this project, especially Norbert and Susanna, who invited me to work on this project. And thank you for your attention. And I'm looking forward to answering any questions.
Thank you very much, Paul. Yeah, were there any questions?
Thank you for the very nice presentation. It's Aude Remaux from INRAE. I'm just curious about the samples that you've collected for the study, even though the results are coming soon. So for tonsil and lungs, it's post-mortem sampling. Do you have any sample that you can compare before and after the challenge, for instance, nasal swab, or maybe also like oral swab? Because I know that lung microbiota can also come from the mouth in some species. Is it the case in pig? Is it proven that the lung microbiota also come from microaspiration from the mouth? What kind of samples are you collecting, and can you compare before after challenge? That's my question.
Right, I'll go a few slides back. Yeah, so maybe I didn't explain very clearly, but the sample collection is indeed at the state of necropsy in this last day. And there are more samples collected, but the focus is first on this this last time point due to prioritizing our research questions. So these are samples from both the upper respiratory tract, so in the nasal and lung and tonsil samples, but also the lower respiratory tract. Now, from the lower respiratory tract, it is known that it's less clear what the exact composition is in that regard. So the upper respiratory tract has a real microbiota that is known to create this this balance, this equilibrium. But for the lower respiratory tract, there's still a bit of a discussion whether this is more of a fluctuation or this is really a steady composition. So yeah, depending on that relation, I think it's important to really compare them side by side and have all of this information. Did I answer your question with that, or not entirely?
Yeah, you answered. I was just curious if also you collected some samples at day 0, just for instance, nasal swab, but I understood that you have more samples at day 21. So.
Yeah, there are more samples, but based on the amount of time, budget, and so on, it might not be a part of this this study to also look into these, but there's at least more samples than visualized here.
Yeah, you have to prioritize it. Yeah, okay. Thank you very much.
Were there any other questions? No, all clear. I was just gonna. We're due a break now for 20 minutes, but I think yeah, if everyone's happy with that, just so we can get through things. Yeah, okay, cool. And then we've got the last 2 talks.
Yeah. No trouble. Concentrate. You're not here to play games, you know. I think I just need to get going. You got a big stick. Sent Kate over here. The last one. Yeah, I'll just get started. Thank you all for remaining till the bitter end. As always, the best is always at the end. So well, I'd like to think so anyway. I'm going to carry on in the work package 8 theme, which is centered on the development of novel analytical tools and reagent. I think this is a really important work package, but because it's designed to exploit and to get the maximum out of any animal infectious disease study that we carry out. Obviously, as the 3 R's are really important, and if we are using animals in infectious disease studies, really we need to get the maximum amount of information out of those. There are vast numbers of tools and reagents available for rodent and human studies, but as soon as we start getting into livestock and indeed wildlife, the options become much less. So part of this work package, and indeed work packages 7 and 9, have developed and are improving the range of tools and reagents that are available to people working on livestock and indeed wildlife disease, infectious disease. And what I'm going to talk about is something called the immunological synapse, which you may not know much about the immunological synapse, but it is, you're all working within it, because it's the ability of the immune response to identify what's foreign, what's pathogen, and then to mount an appropriate adaptive immune response to it. So central to that immunological synapse is the recognition of foreign antigen and how the immune response recognizes something that's foreign compared to the range of self-antigens and other antigens that it is exposed to. And involved in that is a series of molecules called the major MHC molecules, or major histocompatibility complex molecules. And those present small parts of the pathogen to cells of the immune response, or lymphocytes. And the lymphocytes recognize the combination of MHC, self-MHC molecule, foreign peptide through the T cell receptor. So central to the immunological synapse is presentation of molecules via the MHC to the T cell receptor. And I'm going to talk mostly about the MHC in this presentation, and I'm going to tell you why the MHC, why we should be interested in not just the MHC, but genetic diversity within the animals that we are working with. We work on outbred animals. We don't work on inbred mice very much. And the reason people work on inbred mice is because they're all the same. They're the same MHC, the same genetic background. And it means that the responses they look at are much cleaner, but it's not the real world. The real world that we work in is we have outbred animals that are genetically different. Some are more different than others, but generally there is a huge amount of genetic diversity in the animals we work with, and trying to identify how much diversity is in the animals that we're using in infection disease studies and in wildlife populations, I think is really important. So I'm going to talk about that. So we can have the next slide, please. So why are we working on this? So we, as I've already said, we need tools that support the development of vaccines, development of disease control methods. So I've mentioned the MHC, T cell receptor, and B cell receptor, and that's what I'm going to focus on now. I mentioned tetramers at the bottom there. Now, tetramers are really useful reagents, and it allows you to identify antigen-specific T cell responses. In order to do that, you need to have an MHC-defined background, because the tetramer is diversity is linked to the host diversity, so they have to be matched. Okay, if we can have the next slide, please. So this task 8.2 is the development of high throughput genotyping technologies targeting the MHC, TCR, and indeed the B cell receptor repertoire. And we wanted to do that. First of all, this task 8.2 is divided into 3 different subtasks. First of all, the development of high throughput MHC class 1 genotyping technologies. So that focuses on the class 1 MHC genes. And then the development of high throughput TCR genotyping technologies for cattle and swine. And then the development of high throughput BCR, or the B cell receptor genotyping technologies for swine, chicken, and fish. And the various partners involved in this, as ourselves at Morden and Roslin, Tim Connolly at the Roslin Institute, the TCR for cattle and swine is the Roslin and ANSES. Daniel Dorey has been working on that, and the swine, fish, chicken, and fish part of the B cell receptor has been Daniel at ANSES. So if we can have the next slide, please. Okay, so what's the major histocompatibility complex? It's a cluster of genes that are found in all vertebrates, or pretty much all vertebrates. It provides the context by which the immune response recognizes foreign antigen and presents that antigen to T cells. CD4 positive T cells generally recognize class 2 MHC linked to small fragments of exogenously derived antigens such as parasites or bacteria or anything that's floating about. Whereas the CD8 positive T cells will present via class 1, will recognize class 1 and antigens that are within the cell. So it's generally surveys intracellular pathogens, viruses, protozoa bacteria, anything that's within the cell. If we can have the next slide, please. So the key features of some of the genes that code for the MHC molecules are shown here. They're polygenic, so there's multiple genes with immune-related functions. The key is that they're polymorphic, so there are thousands of different variants of each of the genes within the animal populations that we're looking at. I've put the HLA there, which is the human MHC. If you look at the number of class 1 alleles that have been identified in humans, it's 25,000 different variants of the class 1 alleles, which is a huge variation. That's just in people, which are a pretty dull, fairly non-diverse bunch of animals. Whereas the animals that we look at within livestock will have similar amounts of diversity, if not a lot more. There's codominant expression of these genes, so you get both haplotypes. The maternal and paternal haplotype is the genes are both expressed. There's high level of linkage disequilibrium across the MHC, so they're quite tight together, and generally there's not a lot of recombination that goes on there. And within, certainly in livestock that I've been working on for many years, there's variation in the sets of genes that are inherited from each parent. So some haplotypes will have 1 or 2 different MHC class 1 genes, whereas other haplotypes will have up to 5 or 6 different MHC class 1 genes. And why that is and what the functional significance of that remains to be determined. The next slide, please. Okay, so the methods associated with pretty much all this is basically an amplification from RNA or cDNA. So you're looking at repertoires. So you amplify and then paired end sequencing. So you're amplifying using barcoded primers. So you can go back to the individual animal and everything's pooled. So you can do hundreds of animals all at the same time. Paired end sequencing. You target all the different genes you want to target, so class 1 and all the class 2s all are done at the same time with different sets of primers with the same barcode for each animal. And then some clever bioinformatics untangles the whole thing at the end. So you get then a range of genes for each animal, or each set of T cell receptor genes, or each set of B cell receptor genes that you're interested in. If we can have the next slide, please. So I'll just go and give you some examples of what we've been doing. So the development and application of high throughput genotyping technologies for sheep, which is what I've been specifically working with. So the workflow is essentially you get as many sheep as you can. We work within a single breed, but take flocks from a range of different sites, so you get all the variation that you can find just within one breed, and then you can expand it to different breeds. You would take a blood sample, collect about the buffy coat or all the white blood cells, generate RNA, first strand cDNA, and then you amplify with gene-specific barcoded primers linked to each sample. The amplicons for each animal are then pulled together into one big library, and that's then sequenced and then goes through the bioinformatic pipeline. So for 200 black-faced sheep, of which we then selected 100 for sequencing, we identified over 132 different MHC class 1 transcripts. So these are transcripts that are have high levels of sequence variation. That's just within a single breed, within 100 animals within that one breed from 4 different flocks. and that's just MHC class 1. So those different transcripts can be split into 38 haplotypes, so combinations that are inherited together from each parent. So 26 of those haplotypes were identified in more than one animal. So they're validated, they're real. For this, we used 3 independent sets of primers for the MHC class 1. So that allows us to get some quantitative difference based on the read frequencies, because the read frequencies for each of the sets of primers were remarkably similar. So if you just got one set of primer, you might think, well, it's just biased because of the primer, the individual sets of primers. But if you've got 3 sets of primers that target different areas of the same gene, then you can get some degree of confidence that the read frequency is related to the transcript frequency. So we've got good transcript frequency data on that. So that's really nice. And we can divide the haplotypes into high, medium, low, and very low expressed transcripts. Now this is just in peripheral blood, but there'll be all the different cell types that express MHC class 1 in peripheral blood. So for each of these 38 haplotype combinations, we can identify usually between 1 and 2 genes that are very highly expressed. And that's what we're assuming are the classical, or what we consider classical MHC class 1s. They're highly expressed, highly polymorphic genes that will present peptide antigen to CD8 positive T cells. The medium ones probably do the same. Again, they're just, for whatever reason, they're always a medium expression, and why that is, I don't know. The low and very low, we assume, are the non-classical class 1 genes, the genes that have different, slightly different functions, but highly specialized functions. So, and you know, the difference between the high and the low is enormous. You know, so some of the transcript, some of the haplotypes that just has a single class 1 gene, you know, the numbers you can see there in brackets is the percentage of all the class 1 reads linked to that one gene. So if you look at the second one down there, haplotype 5.1, nearly 87% of all the reads were that one. And that the N number in 20 different animals, that was identical. So it's not just one animal, that's in 20 animals, they only have a single highly expressed class 1 gene. And then a number of low ones that just trickle along at the bottom. And then if you compare that to the one at the bottom, which has got 2 highly expressed, and then 1, 2, 3, 4 low and very lows, the numbers of different genes in each have these haplotypes is quite extraordinary. And of the next, so you can do phylogenetics on these and try and link the clusters with individuals. So you think, well, all the highly expressed genes will cluster together. So it's presumably there's 1 or 2 highly transcribed gene loci in sheep, and then you've got the medium ones tend to cluster together and the very low ones cluster together. So it actually splits in between for different sequences can be aligned to what we think are the different genes. Then you can look at the haplotypes, variation between the different flocks, and the frequency of the haplotypes within the breed itself. And as I've said at the start, this is just one breed of 100 animals, and you can see the variation in just the class 1s. We have done exactly the same for all the class 2s and generated complete haplotypes for these linked. So you've got the class 1 genes and then all the class 2 MHC genes linked together. So, and we have to thank Dipali at the Roslin for doing the clever bioinformatics. She's designed the pipeline. So you do the sequencing, you just say, Dipali, sort that, and she'll go and run it through the pipeline, and it spits out at the bottom all the genes and the haplotypes, and it's really lovely. Have the next slide, please. Exactly the same thing has been done for cattle, and this is Tim Connolly at the Roslin Institute done this. So again, he's been looking at for what's presented here is from 600 African cattle from 1, 2, 3, 4 different breeds, and it's essentially showing how much variation there is in there, but he's also linked the class 2 genes to the class 1. And you can see some on the right-hand side there, you can have a look at this, the recombination within the different haplotypes. And some haplotypes with MHC class 1 and then the two MHC class 2 genes is very tight. There's pretty much a straight line across. But some of them, there's lots of branches going all over the place. So you've got a different set of class 1 genes linked to a class 2 DRB gene, and then linked to a different DQ gene. So there is recombination within the MHC region in cattle as well. And that's much the same in sheep as well. Can I have the next slide, please? So anyway, so just gives you an idea of the amount of variation there is in a relatively small number of sheep and a reasonably large number of African cattle. Very much a similar type of thing looking at the T cell receptors. The TCR is quite a complex rearrangement of genes gene segments within the genome that recombine into to produce a single transcript that's then translated into one of the T cell receptor chains. And most of the variable parts of that are short. The variable parts encode the specificity of the antibody. So and different sets of V, D, and J segments recombine to produce that variable part of the antibody. And each T cell and each T cell clone will have a single set of these genes. So you can get an idea of the repertoire of T cell receptors by sequencing them all. You know if it's a really tight, if you get only a small number of different T cell receptor sequences, or you get a very broad range. You know, there's a lot of different T cells or T cell receptor being used in whatever immune response you're looking at. Can I have the next slide, please? So this has been done mostly at the Roslin, but ANSYS has been involved in the pig side of things. So Tim's been very successful in designing and validating the primers for the T cell receptor alpha and beta chains, and he's also done gamma and delta, TCR gamma delta chains as well. He uses a range of different sequencing methods, but the PacBio is the latest, so that's been working very well. And indeed, he's published one or two papers on the T cell, the T cell receptor gamma and delta, and I think he's in the process of doing the alpha beta as well. So that works across cattle, sheep, pig, and goats. So that this is the same technology works in certainly all these species. Can I have the next? So this is from Daniel. I mean, it's essentially just what I've been saying. He's been working with swine, chicken, and fish using similar technologies, and has spent a lot of time working up the methods, choosing which sequencers to use, choosing which primers to use, because to start with, there was very little information certainly on fish and chicken. So he has, I say, spent a long time getting the methods up and running. Can I have the next slide, please? And he's been focusing on one area, which most people focus on, which is the CDR3 region, which is formed between, you get the data from the V, D, J regions of the transcript. So you get a lot of information on a relatively small piece of sequence. Can I have the next slide, please? So he's been involved in primer selections, choosing the source of the RNA, because where do you start? You know, you can get RNA from single cells, or you can get RNA from PBMC, or using different systems like the Leucoloc system, or just whole blood. So what's the best source of RNA? So he's eventually chosen PBMC as the best after doing a number of studies just to check which one gives the biggest range of diversity. He's chosen the MySeq platform over the Ion Torrent. I think at ANSYS they originally had the Ion Torrent on site, and so that was what they started with. And then since then we moved over to the MySeq platform, and been looking at then data analysis pipelines. There are a number that are out there anyway. So he's been looking at the IMGT and then high V-Quest. I'm no expert on these, so I have no idea which ones are good or which one's bad, but Daniel's been testing a bunch of these. And then he's been applying these to a number of different experiments. Pigs infected with porcine endemic diarrhea virus was one of the first ones that he was testing the methods on, and he was comparing the CDR3 region and the repertoire of B cell receptors before and after infection at day 7, and just looking to see what the difference is in the repertoire of B cell receptors that had been using. And some of the differences in the usage of VDINs before and after infection, he was able to identify, but there wasn't any difference in the usage of the D and J genes, and he couldn't find any common antibody specificity or CDR3 sequence that was common to that specific immune response. But the analysis of this is ongoing and using different time points and different infections. So it's an ongoing process. Can I have the next slide, please? And he's been doing very similar things, at least starting the process to look at the trout B cell receptor. And certainly it appears to be the RT-PCR seems to be more complex, and he's been having difficulties in the choosing primers and that give a reasonable range of B cell receptor diversity. Can I have the next slide, please? He's been looking at the spleen from infectious hematopoietic necrosis virus infected trout using different strains of I think virus with different virulence points, again at 2 time points. So you can see how these type of experiments can expand to a huge level. And when you're trying to work up the technology, it's not easy. Anyway, had some data on infected versus uninfected, and the selection of PCR parameters clearly has to be optimized to increase the number of sequences, because he wasn't getting enough sequences from some of these runs. So again, this is ongoing work. Can I have the next slide, please? And very similar in chicken. Obviously, they've been using very similar techniques to pull out the VD and J regions. He's collected the bursa and spleen from Campylobacter infected chickens at different time points after infection. And again, this work is still ongoing, and a lot of optimization is still going on. So can I have the next slide, please? All that really remains to be completed for the deliverable is for me to get my get it together and finish writing the paper. The paper's to describe the genotyping of class 1 in the sheep. It's 3/4 done. All the bioinformatics has been done. It's just finding a time, because I have to come to these meetings and stand up and talk a lot. Just to sit down and finish this paper and get it submitted. And I think with that, I'll be quiet and happy to take any questions. Bye, Kate. I rattled through that quite quick because I was I know its time is of the essence.
The H5.1 is highly represented, isn't it? It's 20 animal out of 100, it's pretty high.
Yeah.
I mean, can you already associate this haplotype to a specific immunological phenotype?
Not at this moment. We would need. Yeah, no, we haven't. We've got the technology up that we can answer those sort of questions, especially for the high frequency. Yeah, excuse me, the high frequency haplotypes like that.
Would be interesting, very interesting.
Oh, very much so. Yeah.
Thank you.
All this diversity, do you think that it's because the inbred have not been so much selected and they keep the capacity to adapt, or how do you interpret that in comparison to?
I mean, the current theory of the generation and maintenance of diversity across the MHC is that it's there to deal with ever rapidly evolving pathogens. So Yeah, and despite a century of selective breeding for other things, usually production traits, there still remains a remarkable degree of diversity in certainly, well, in sheep and cattle. African cattle are special because they haven't gone through the same intense selective pressure that a lot of European breeds. If you look at Holstein Friesians, they do have less, not surprisingly, especially when artificial insemination using very limited number of highly selected bulls. The problem is you get less and less diversity. So they might be very good at producing a vast amount of milk, but the reason you have diversity within the MHC is it provides a degree of protection against new and rapidly evolving pathogens. So you would worry if there is new pathogens arise because there isn't the diversity in the population to respond to it. Anyway.
Actually, this is my naivety with immunology, so bear with me. But you know, I'm aware that the TCR and the B cell receptor, they undergo mutations within the host, but the MHC not. And I was just wondering.
No, not.
I was just wondering.
You inherit the MHC a set from your mother and a set from your father.
Yeah,
there is a chance of recombination, but it's rare.
So I suppose the question is that is there therefore, we were saying about the diversity of the MHC to account for this diversity in pathogens and evolution, but is it therefore less of an issue for the MHC than it is for the TCR? I'm not too sure how the diversity enables different bindings sort of thing.
You would expect that animals that would normally have a lot of MHC diversity, when you remove that, you start having problems with fertility, because the MHC diversity is linked to fertility, pregnancy, success of pregnancy. Now, whether that's a direct function of the MHC or it's an indirect function, that in fact you're actually become less diversity across the rest of the genome as well. I mean, you can use diversity at the MHC as a proxy for genomic variation. So if you manage to reduce MHC diversity, you can bet your cotton socks you've got a lot less diversity across the rest of the genome. I mean, we've tried to inbreed animals to get MHC homozygous animals, and indeed Frédéric Lontier spent many years developing lines of sheep that were by inbreeding year after year, and eventually they were so infertile they couldn't keep them going. But there have been a fabulous resource for looking at regions of the genome that are normally highly diverse, because they weren't diverse, they were homozygous. So some of the initial work, we used RNA from those homozygous pre-alp sheep lines, and they were all homozygous across the MHC, which makes life a lot simpler. Yeah. So yeah, reduce diversity at your own peril. It's all gone very quiet, which is a good sign. I will shut up and
thanks very much.
And move
Thanks,
on.
Keith. Yeah, I've got the last one now for. Oh, lovely. Thank you. Okay.
You can just.
I'll click. Yeah. Okay. Oh, lovely. Thank you. Okay. Thanks very much. My presentation's the last. I'm just going to very briefly summarise, because I'm conscious of the time. Very briefly summarise one element of the work that we've undertaken at Pirbright in this work package. And so this was the deliverable, or the task and deliverable within which I'm presenting this work is the examination of the viral swarms, the viral quasi-species analysis by NGS move that there. This has been published. The first, there's two publications that have come from this. The first is to define a pipeline to enable these low frequency variants to be detected within viral swarms, and the second was to then use that pipeline to assess an evolution of FMDV using an in vitro system with pre-existing immune pressures. So we added some subneutralizing antibodies to it. I've got this on here, not so I'm going to read it out, line for line. As I say, that the papers, both of these are published, but I just wanted to very briefly discuss the approach. So the approach for the pipeline was to actually use artificial synthetic sequences and basically spike in different proportions those different sequences to see what the limit of detection was. This is very basic terms because as I say, this isn't my work. To then use that to develop the pipeline to see what the cutoff values are, and to enable us to be able to then define which of the variants that we see with the deep sequencing with the NGS are real and which are likely to be due to artifacts. This is a diagrammatic representation showing these different showing the different levels of DNA and RNA that are used. RNA high is there's a spike of high concentration relatively of RNA within a sample. The medium's medium, the low is low, and the DNA the same. So we used RNA and DNA artificially created to spike into these samples to then sequence. And the take-home message from here is that actually with DNA high and low, we still have some relatively, and on this graph, by the way, the y-axis, the higher it is, the median MCC, the higher that is, the closer that is to 1, the lower the variants that we can detect are in there. We can see the take-home from this is that with low RNA levels, we're less likely to be able to pick up these variants at a specific frequency, largely thought due to the RT process, the introducing errors into those annulates. And the second bit of work, as I said, so that was the first part, and the second part was then to use that pipeline that was developed to then to try and assess the use an in vitro model of immune pressure to see how if we can use that pipeline to assess the mutation, what effects a pre-existing immune response or immune pressure has on FMDV evolution. So very briefly, what was undertaken was there was in vitro passage of virus in the presence of FMDV, in the presence of subneutralizing antibodies, and that was then passaged multiple times. That was the in vitro side, and then there's also an in vivo. There was an in vivo study as well that was undertaken, which actually had animals with subneutralizing levels of antibodies to FMDV were then infected, and then lesions from those animals were harvested, and then again deep sequence to look at the what effect that had on the variants within that population. So just to focus on this first one, the in vitro model, as I said, we had infected these LFBK alpha V beta 6 transfected cell lines with FMDV at a low MOI, and then that was this was then passaged with subneutralizing antibodies, and then these sequenced. And actually, but one of the things that was the decision on when to sequence was this reduction in time to CPE. So the theory being that if you had viruses that were being subneutralized by these antibodies, the time to CPE should decrease as the virus escapes that polyclonal neutralizing antibody antibody present in the in vitro system. And when that became stabilized, that was when that part of the passage is when there was these lysates were sequenced. So this just shows this just shows a graph of this this approach. The time on the x-axis obviously is in hours. The CPE on the y-axis, and we can see with every passage. So it starts off orange with every passage. In the presence of subneutralizing antibodies, we saw a reduction in time to complete CPE. So the virus the theory is the virus is mutating to escape the polyclonal antibody response. And upon sequencing, we saw sequence the starting virus and then various other viruses. These are viruses actually from the challenge study, in the in vivo challenge study. And again, in here there were various mutations noticed, both synonymous and non-synonymous, but with the amino acid changes in particular highlighted here, that were in areas of known epitopes. So a way of being able to, and that was using this pipeline that was developed previously with a view to be able to see which, in these certain situations and these certain subneutralizing antibody responses, which were the important epitopes that enabled potential virus escape from those, which is obviously an important element to understand in the context of vaccine development, for example. And in this in vivo study, we used animals that were had a pre-existing subneutralizing antibody response from a previous study where we looked at a polyvalent vaccine. The animals were not protected, but there were some neutralizing antibodies in that. In here, this is just a very summary graph of the mutations that were present here. So the percentage frequency in these is along the y-axis, and the nucleotide positions along the x-axis with the polyprotein along the bottom. And highlighted on here in these different symbols show the different, each different type of symbol shows a different animal, and we can see how these are spread throughout the genome. So it's not just the in P1, which is VP not just in VP1, which is thought to be one of the most important proteins present for antigenic variation and protection, but actually it's present throughout, including in the leader, so the protein that's not present in the capsid of the virus. So there's further work to be done to understand what the significance of these are, but this it was a proof of principle to show that this technology can be used both in vitro and in vivo to detect these changes. So and that was very brief summary of the work that was undertaken. But as I say, I'm conscious of the time, so thank you very much. And if there are any questions. Sorry, yeah.
Ryan, thank you for your presentation. Maybe this assay could be very useful to predict the hot spots in the foot and mouth disease that are more variable. So to be ready for the selection of the vaccine strain that match better for a potential escape viruses from in the nature, that like the virus is highly variable. With the time, we have to perform these matching experiments to check if one strain is capable to induce the correct immune response to be protected by the field samples. So this in vitro assay could be used to predict this and to be ready, or?
Yeah, I mean, I think so. As I say, it wasn't my work, but I think that depending upon what serum's being used as well. So if you've got a certain, using serum from vaccinated animals, and then using the different field viruses circulating within that pool, for example, I guess you could do that in vitro using multiple isolates with that same immune serum to see if you had convergent, or the same areas that were being affected. I guess that could be done to predict that, in those areas, as you say. So yeah, I think so. Yeah. Okay. Well, thanks very much. And I just wanted to say that, thanks to everyone. This is the first time I was on an EU grant, and when I started, I really didn't know too much about how it all worked. But I just wanted to say it's been really nice meeting everyone, working with everyone, putting faces to names, understanding what facilities are around the world, and actually through this we've had lots of conversations outside of the the tasks and the deliverables, and I think that they'll continue in different areas, as we go forwards. So I just wanted to say thank you, and it's been a pleasure working with everyone. So yeah, thank you.
Okay, so conclusion for the final meeting. That would be now the moment for nostalgic speech, carefully drafted since weeks, but then it's late. There's only perhaps a dozen of people still listening, and to tell you the truth, I haven't prepared anything, so I keep it short. And there's no need to become nostalgic, actually, because okay, the VetBioNet project funding period will expire by the end of the month, but I feel that VetBioNet as a network is still very much alive, and I'm actually convinced that we will meet soon again under the banner of the VetBioNet ERG. But still, I just want to thank everybody also for all your commitment to the project, to the network, for your hard work. And then finally speaking for myself, for Mabel, but also for Frédéric and Anthony and the previous project managers, Joselle and Oriana. I just want to say that it has been an honor and a pleasure to serve you in the coordination and management team. So thank you very much.